So before starting, I just wanted to thank all of you for being here and coming from very far away. So if we could have the first slide. Yeah. So basically, we are now at the fifth week of this program about advanced asymptotics, modeling, simulation, extreme statistics, and the application to data analysis in cell biology. So just to remind you, the first week was about statistical physics, extreme statistics, and modeling. The second week was about match asymptotics, how to analyze and to get a solution, explicit solution or asymptotic solution of PDEs for the respective models. The third week was about multiscale modeling and simulation. Last week was about synaptic transmission and the modeling and simulation. And this week is about this reverse mathematical method. And next week will be about nucleus organization and how we use modern methods of statistical physics to reconstruct chromatin. So this week, reverse mathematical methods for reconstructing single molecule dynamic in cells was organized by Can Dowduck at UC Berkeley, Nathanael Ozzi at Pasteur Institute, and myself. So first of all, I would like to thank the center here. We are hosted by the Dojoji Center for Mathematics, which give us the opportunity to organize this entire program, and in particularly to have this room where most of the talk, all the talk will be video recorded. Stefano Marmi is the director of the center, so I would like to thank him, and Luigi Ambrosio. And finally, I would like to attract your attention if there is any financial or administrative issue that you are facing, please contact Katarina Delia. But this week, we will have Antonella, the two secretary, Antonella, and Luisa. So I think here we are week number 5. So this is Luisa that should take care of us. Is that correct? So Luisa is here. So if you have any questions, anything about the hotel, just talk to her. What I propose is that on Wednesday, we are going to try to have the free afternoon on Wednesday. So we are going to move some of the talk of Wednesday afternoon probably tomorrow. Just this is a little change in the program, so this will allow us to have Wednesday afternoon free. And I think this is important, especially if you want to enjoy a bit the city. All right. And basically that's it. So I want to welcome all of you, and Can will chair the morning session. And I'm looking forward to this week. Thank you.
Does that mean this program is not correct?
It was correct 5 minutes ago.
Okay, good morning, everyone. I also want to add that for the dinner on Wednesday, I sent a form to fill. So if it's useful for us to have a head count, and also if you have any dietary restrictions, it's also good for us to know. So for those who haven't, if you can please fill the form. And so I guess we will start with Erwin Peterman from VU Amsterdam. And he's going to talk about some, I need the program, the exact title, because it will be, so single molecule view on intracellular transport in vitro and in vivo.
Let's see if it works. Yeah. Good morning, everybody. I'm a bit nervous. This is my first mathematics conference. How do you call it? My last math exam was more than 25 years ago, I think. So I'll do my best. So I will talk about stuff we do on intercellular transport. I will start with discussing stuff we do in vivo and also give you, tell you something about the stuff we've done in vitro. And I'm from Amsterdam, and there are many nice things about Amsterdam. But one of the most signature things, at least for me, is that you travel around by bike in the city. That's by far the fastest. If you look in the basement of my apartment, I have 5 different bikes. It's not completely the current status anymore. There is some turnover, but never mind. This is the bike I use to cycle to the university. This is my girlfriend's bike. This is a folding bike I can use to take on the train. This one I use for a spin in the weekend, a fast spin in the weekend. And this one if I have some bigger plans in mind for going to North Cape or something like that. And you see, they're all very similar. They all have 2 wheels and 2 pedals and 1 handlebar and a seat. But they are quite differently constructed because they serve different purposes. And if you look into our cells, then we see that transport is also very important in our cells, and that nature has invented also many tools to transport stuff around. Most remarkably, I would say, the motor proteins, of which there are 3 superfamilies. The myosins, most well known for muscle action, but quite a few of them are also crucial in intracellular transport of stuff. The kinesins, my personal pet proteins, they're the smallest motor proteins. I think there is about 42 different ones in our genome. They act on, in contrast to myosin, which acts on actin, they act on microtubules. Of course, dynein also acting on microtubules. And sort of the question we have been asking in the lab is how do these different motors together drive transport in cells. And stuff we have done many years before is to do in vitro assays where we use microtubules that we've isolated from pig brains, and we reconstruct them and attach them to a glass surface. We add fluorescently labeled kinesin. I can write this down and say it in 1 second, but this is of course the months of work. We make sure that they're fluorescent so we can track them with single molecule fluorescence. We throw them in our turf or otherwise microscope, and we add ATP as a source of energy, and then we can see the motor proteins walking, and under the microscope it looks like this. And from these kind of assays, and I'm showing you here single molecule fluorescence assays, but of course optical tweezers assays have learned us also a lot about how these motor proteins work. And we know stuff like that motors walk on the order of a micrometer per second, that they do not walk infinitely long, but that they make sort of hundreds of steps before they release. What we also know is that kinesin works on its own, as a dimer, I should say. And yeah, what we call this behavior processive. Kinesin makes many 8 nanometer steps or 16 nanometer steps. Depends from your point of view, a longer microtubule before it releases. Fantastic, but if you think a little bit longer how kinesin should work in a cell, the situation is a little bit more complicated. First of all, kinesin might be transporting huge cargos. Second, the cell is a rather crowded environment. There's many ways you can look at that. First of all, of course, the microtubule might be densely occupied. Not all binding sites might be available because of other motor proteins, but also because of other proteins binding to it. But you can also imagine situations where multiple cargos are pulling on the same, multiple motors are pulling on the same train. What happens then? Of course, in the most extreme case, they would be pulling in the opposite direction. What happens there? And you could imagine that the cell has already had a couple of million years to think about this and find good solutions regulating switches from being a transporter to cargo. I've been fascinated the last years by these kind of questions. How do motor proteins, or how does life solve this? And I will give you 2 examples of what we have been doing over the last years. I will end with giving you an in vitro assay where we study the behavior of motor proteins at very high densities to see how this traffic jam kind of behavior affects motility. And you would expect that if you buy a new computer that it goes faster, but for some reason, not with PowerPoint. But the main part of my talk, I will be talking about in vivo work on interflagellar transport, a specific transport mechanism in C. elegans. This transport mechanism takes place in all cilia, and cilia are crucial organelles in almost all eukaryotic cells. We love to look at C. elegans because C. elegans is fantastic. It's transparent. It grows very fast. What is it, in a week or something like that? It's a hermaphrodite, so it basically clones itself, copies itself in 300 identical copies. Many, many reasons. And for Dutch law, it's not a test animal, so we don't have to fill in too many forms. And what we specifically look at are the so-called chemosensory cilia that are connected to dendrites, so they're connected to neurons, and that are involved in chemotaxis, namely in sensing salt, pheromones, or also bitter compounds of plants, and make the animal react to that. Okay, people had been studying this system already for quite a while, and there were some interesting things that struck our attention. So first of all, this is sort of a schematic view of a cilium, very schematic. A cilium is basically a little antenna on top of a cell. It consists of a bundle of microtubules called an axoneme. In C. elegans, this is about 8 micrometer long, and it's enveloped by a membrane. So this is the rest of the cell, and then in C. elegans, this is on top of a dendrite. So the interesting thing is that transport takes place in so-called trains. So we have multiple motor proteins that bind to so-called IFT trains. These are protein complexes that cluster in copies of multiple tens, about 100 in fact, that transport together in sort of a conveyor belt way from base to tip and back again. What my collaborator John Scuola had found about 15 years ago is that there is an interesting, something interesting going on. There are 3 motor proteins involved in the transport, 2 kinesin motors. I might confuse you hopelessly with the names, but I will not mention them now, maybe later. 2 kinesin motor proteins together take care of transport from base to tip. While there is one specialized dynein called IFT dynein that takes care of the transport back. And the funny thing is that the 2 kinesins together drive transport at sort of an intermediate velocity in the beginning of the cilium. And a little bit later, the velocity increases because only the fast one sort of survives. People could not really see clearly what was going on. There is a structural change sort of halfway in the axoneme, and people thought that would be the trigger.
Quick question about that.
Yeah.
So in the picture it looks like In your picture, it looks like the two parts of the heterotrimeric kinesin 2, the two motor monomers, are different from the one that forms the homodimeric kinesin 2. Is that right?
Yeah, that's right.
Okay, good.
Yeah. And the question we have asked the last years is, how do these three motor proteins work together and bring about this fascinating transport? I will focus on three things, or four things. We'll see how it goes with time. First, on the cooperation of the two kinesins. I will also briefly tell something about stuff we have learned from the IFT dynein, the backward transport. I will focus a bit on what happens at the tip, where we really get this sort of direction switch. And I will also tell you something about recent work, or at least recently, very soon to be published. No, has just been published work where we, combine studies of ultrastructure towards study of motor behavior. Okay.
Could we just ask what IFT is?
Intraflagellar transport. Rewind in history. Bacteria have flagella. Other animals, I should go further back. Anthony van Leeuwenhoek, we all know, I hope, he discovered flagella cilia for the first time, and he saw them in, some unicellular organism. People have seen them in bacteria. People thought for a long time that that was the same organelle, but of course it's not the same organelle. They don't have anything together. So that is why people still give the same name to bacterial flagella and eukaryotic flagella. I tend to use the term motile cilia for eukaryotic flagella, but probably I will be shot by people working on chlamydomonas, for example. If I say that, so flagella is synonymous to cilium for most people. Okay, so we attack this problem from our expertise, which is mostly in data analysis, maybe a little bit in data analysis, mostly in single molecule fluorescence microscopy. So one of the first things we started doing is to start dive into biology, because we noticed that people before us had not really taken the most modern methods to make fluorescent worms, because if you look at a worm, it's transparent, you don't see anything. You want to make the proteins of interest fluorescent, so you use genetic modification. We used endogenous expression to really try to make sure that that we get natural expression levels. We used this Mosky method before. We're now, of course, using CRISPR-Cas because that's easier. We use our own built epifluorescence microscope with very high sensitivity and flexibility. We put a lot of effort in automatizing data analysis, and also in getting single molecule sensitivity and approaches to really see single molecules walking around in the living worms. So if you take one of these worms, and in this case it's not a few months work, it's really, the first one was 1.5 year work. We can do now a bit faster, and you put it under the microscope. This is what you see. Time 1 micrometer distance. What you're looking at is EGFP tagged on IFTB, and IFTB is one of these train proteins. So I envision the transport a little bit like a cargo train, and good old American cargo train, where you have this long train consisting of carts, to which the cargo loads, and there is multiple engines in front, or sometimes even in the back, that drive the transport. So IFTB is one of the cargo carts. This is the base, this is the tip, and as you can see a lot of stuff moving around, but it's overlapping quite a bit. Your eye is a bit deceiving in seeing a lot of transfer from base to tip, and a bit less but you can see it in the other direction. And I can tell you later on why. So how do we analyze this kind of information? We cannot do single particle tracking on it, it's too crowded. So what we do is we make chimegraphs. I don't know if the audience knows what chimegraphs are. Basically, we just draw a line along the cilium and plot the fluorescence intensity along this line for each movie frame under each other. If you get a straight line, it means that stuff is moving at a constant velocity. If it's a vertical line, stuff is standing still. What you see here nicely, so we do something else. We do Fourier analysis to color code for directionality. So in green you see the forward transport, anterograde transport, and in red the retrograde transport. What can you immediately see? First of all, that there is about twice as much retrograde transport. Intensity is lower though, so that is probably why our eyes are deceived. The backward transport appears pretty straight, relatively constant speed, while the forward transport really has these slanted lines. So we can analyze this. We spend a lot of time on automatizing analysis of this, so we can really automatically trace these lines, get intensities out, and get slope out. Slope is of course related to velocity. So one of the things we do is to plot the velocity for many cilia, many of these traces in chimegraphs, and we can get a nice average of how the velocity develops. It starts at about half a micrometer per second at the transition zone, then it slowly increases after leaving the beginning of the cilium, reaches sort of at 3-4 micrometer, it sort of reaches maximum velocity of about 1.2. I should add there is a lot of variability there. We can also look at the motor proteins, and what you see here is the forward transport. Only the forward transport, and I used here a worm which has the two kinesin motor proteins fluorescently labeled with different colors. In green, the kinesin 2, the heterotrimeric kinesin 2, and in magenta, the OSM 3, the faster motor, the homodimeric kinesin, Arabic 2. And you see a lot of interesting stuff in this chimograph. First of all, you see that lines start green and then slowly turn, this region, turn into magenta. But they're sort of more or less continuously moving. So what we did the same analysis again.
Just a quick question as well. Yeah, for question, the microphone will come.
Okay, thank you. So my question is, do you mean this recruitment of the magenta type as well then?
Sorry,
so there's actually a recruitment of the magenta type?
I will come back to that in a second, what is going on. So what we see is, if we just look at these bulk assays, we can really count the intensity and calibrate it to numbers of GFPs. And what we see is that we start with sort of 50 or so of these kinesin 2s and only 10 of the OSM 3s, but then after sort of 1.5 micrometer, the kinesin 2 number appears to drop, sort of 0 after 4 or so micrometer, while the amount of the other motor increases gradually. That's what we can say from these experiments. So to get more insight into that, we have to look at something else. But before doing that, what we did is we sort of tried to correlate velocity towards motor ratio to see if there is something interesting there. We have both that information. So what we can do is we can calculate the OSM 3 fraction and plot that against the train velocity we measured. Our data are these black points. And if you would assume a stupid model where the two motors basically contribute equally, so I have 100 motors then, and the system takes 100 steps. If 50 of these steps would be taken with the velocity and the properties of the one motor and 50 of the other, then we would get this blue curve. That's clearly not fitting. If we assume that OSM3 contributes about 10 times more than kinesin 2, then we can get a better fitting model. Very simple modeling. Steve Block did on basis of optical tweezers experiment recently a little bit more advanced, maybe, yeah, a little bit more advanced modeling approach to try to understand this behavior. But to really get insight into exactly the question what you asked, what is going on with these motors? They cannot arrive from nowhere. We thought, let's try if we can track individual molecules. So what we did, I should not show you what it looks like, but what we did is we used two approaches. One approach is that we just photo bleach and wait till enough of the GFPs have gone that we reach a single molecule level, which in fact works really well. Or we use a photoactivatable version of GFP, which we can excite with, which we can sort of switch on and in a more or less regulated way, such that we really arrive at the single molecule level. So this is some of the movies we got from kinesin 2, and yeah, I always have to tell myself this is really amazing. You can see one single protein doing beautiful acrobatics in a multicellular organism. That's what our detectors, that's what our lasers, that's what our fantastic optics can make work. And it's in fact relatively easy. We can do this routinely. And we can track these individual spots. Now it's possible. The density is much lower. And you see two molecules being tracked. And you see from both of these molecules something really exciting happening. You see that they make a U-turn. They start moving. Here's the base. The tip is somewhere over there. They start moving forward. Then at some point they make a U-turn and start moving back. What we actually think that is happening here is that the motors, they switch direction. That's what you see. But what we think is that they fall off a train. They're first driving the transport of a forward-moving train. Then they fall off a train and are picked up by a train that is transporting back. We still have to formally prove that, but the velocities make sense in that respect. We see the same we see similar behavior for OSM3, where we also can see these U-turns. So to look a little bit more deeply into that, we looked at the locations where these U-turns take place. And what we see is that kinesin 2, from forward to backward and backward to forward, kinesin 2 really cycles between the base and really just 1.5, 2 micrometers, a couple of micrometers away. So it really cycles in the first part of the cilium, while OSM3 cycles over a much further length of the cilium. What we furthermore saw is that the directional switches are almost instantaneously. So this tells us that the motors, this might tell us that there are hardly free motors that are just diffusing around. If they fall off, they're picked up immediately and transported back. Exception is the base, but good. We also looked at IFT diamine, and just to sorry.
Very quick question. Do they ever U-turn again and become drivers once they've been passengers?
We think so, but this is a very rare event in the sense that the sort of the average duration of a trajectory we can measure is about 20 or so time spots. So we've only seen that very, very rarely, that we get multiple switches.
But you can see particles moving retrograde and occasionally becoming anterograde again. Even if you haven't seen the first U-turn, can you see that they were moving retrograde, now they've U-turned and become anterograde?
We can see the individual switches, but we cannot really track a whole particle or whole motor protein multiple cycles. We wish we could, but there are limitations there. For dynein, let me go a bit quick because, let me go a bit quick. We see very similar stuff. We can see dynein driving anterograde. You have to believe me that I know where the orientation of the cilium here. We can track it. We can also see it being transported by the kinesins in the opposite direction. We see diffusion at the base, and we think we've seen that the same thing for the motor proteins. We really think that at the base, you know, it's just like us waiting for the train at the station. We're just walking around, getting a coffee. It takes longer, getting a Mars bar or whatever, until the train goes, and then we clock together and the train leaves. That's what we think that is happening there. It's searching for partners to leave. And we also see, but to a far lesser extent, we see retrograde, we see directional turns. Far lesser extent, and all over the place than the kinesin motors. So together, this sort of gives us a certain view of intermolecular transport at C. elegans. Trains, and it's sort of a, how do you say it, slight improvement of the previous IDs. So we get these big trains consisting of these IFT particles. In the beginning, it's really kinesin 2, the slow motor that starts the transport. It takes IFT dynein as cargo with it. As soon as we have left this transition zone, this magic zone, where there's many proteins connecting the microtubule with the membrane, and it really forms a diffusion barrier of the rest of the cell and the interior of the cilium, really localized in this 1.5 micrometers. As soon as we have left that region, the kinesin 2s start falling off and OSM3s start binding. We get an acceleration of the motors until after a couple of micrometers in this middle segment, it's only OSM3 that takes over the transport and makes sure that transport goes at maximum velocity towards the tip. Then something magic happens. I hope I can tell you a little bit more about that a little bit later. And IFT dynein takes over and takes with it the kinesins as cargo. It moves with relatively high velocity all over the way, and also picks up the kinesin 2s. And train backbones seem to move in one go, while the motors, and in particular the kinesin motors, really dock on and off trains and modify the velocity, resulting in a concentration gradient, which we can model assuming some simple parameters and assuming these locations where where the switches take place, and assuming the velocity, we can get the actually measured concentrations back. So what does this tell us about the properties of these motor proteins? So we think that kinesin 2 is really the crucial motor for import. So really make sure that trains are formed and can go into to the cilium. They can navigate this densely packed transition zone region, while OSM3 is really the efficient long distance transporter. And for some reason IFT dynein can do everything. It's also much larger. So I would say kinesin 2 is the enduro bike, OSM3 is the racing bike, and IFT dynein really the heavy multi-purpose bike. Good. Let's focus on what is going on at the tip. There we really get this massive directional change. We've been looking at that the last couple of years. So far it was very difficult in the data I showed you to look at the tip, because this is a cartoon of the orientation of the cilia. Cilia are quite straight except for the last 1-2 micrometers. Unlucky us. But what we found is, by doing some confocal studies to really make a three-dimensional reconstruction, that there is one orientation of the worms. So basically the worms are oriented randomly in our microscope. There is one orientation of the worms, this one, where the tips are bending outward, and we can really select this sort of 5% of worms, and there we can really see the whole cilium in focus. So that's what we focus our attention on for this part of the session. So what we did is we again looked at the single molecule level, and I show you mostly chimegraphs here of, in this case, IFT dynein that makes turns at the tip. And there were a few things that immediately became apparent if you look at these chimegraphs. First of all, that these turns do not take place at one specific location. It's really a region of about a micrometer. And that is the same for other components. So this is OSM6, which is a component of the IFT-B train component. And we think that this is due to that this really gives us information on the actual length of the individual microtubules, which for this animal are, how do you call it, the end of the microtubules. There they are single microtubules, and not anymore axonemes that are highly stabilized. And the length of these individual microtubules might be different, so the length of the different tracks might be different. That's what we think, but we would need more proof to show that. Next, what we looked at is, can we say something about what happens at the tip? And what we saw is that in many cases we get these nice, almost shark fin kind of, is that shark fin? I don't know, maybe not. But very sharp transitions where there is basically no waiting time at the tip. But in other cases we really see that there is a pause of a couple of seconds. So to get a little bit more insight into that, we made cumulative probability distributions of the pausing time at the tip for different components. And we saw a striking difference. So if you look at the motor proteins, dynein and OSM3, and key 11, which is a component of the IFTA trained particle, we see that there is hardly any pausing. Median pausing is less than 600 milliseconds, and if there are pauses, they're relatively short. While IFTB, and in particular OSM6, the protein we use to label, there is really a sort of median pause duration of 3 or so seconds, with some pauses really lasting many seconds. So what this tells us is that trains consisting of these 4 components, they need to be taken apart and put together again, otherwise we could not get different pausing behavior. And there seems to be something special about IFTB. To make a long story short, we think what is happening is indeed that the trains disassemble, and we do not know if that is in, how to call it, really that all proteins fall apart, or that it's really individual protein complexes. More research will be needed. But what we think is that IFTB, or in particular this protein we are looking at, needs more time before it can cause a directional switch. There could be 2 reasons for that. One is that it's actively between quotes held at the tip, such that we need a reservoir of it that needs to be at a certain amount, or it undergoes some sort of conformational or whatever chemical change before it can go back, and that takes time, and that is why it is pausing. IFTB pauses might require modification, or it's really held there at the tip. And that is the question that we try to answer in future experiments. Is this really the directional switch, and what can it tell us? We're collaborating for this with people who are reconstituting in vitro the particles and the motor connections.
Yeah, sorry, I think I got a little bit confused. At the beginning,
Yeah, I may be a little bit confused. At the beginning, you referred to the tip as the tip of the cilia.
Yeah.
And I think now you're referring it to the tip of a segment of microtubule, and there's many segments that make up the train.
Okay,
There's two different meanings.
No. Yeah, there's two different meanings, but they are very topographically very close. So it's indeed, I mean here, this is the tip of the organelle, but it coincides with the plus ends of the microtubules. But of course, the plus ends of the microtubules, they might vary a little bit. I should be clear. Plus ends of the microtubules and the tip of the cilium. And the plus ends, the minus ends of the microtubule are there. Plus ends are here, and this is also where the tip region is. I hope this clarifies. Okay, I'm looking at the time. I'm going to skip this. No, yeah, maybe I don't. I'll do it very quick. So one of the cool things about being able to do single molecule tracking is that basically you can use it to beat the diffraction limit in fluorescence microscopy. So this is, in black you see basically the information we get from a couple of hundred trajectories of motor proteins or IFTB proteins in a single cilium. And you see, no, yeah, it's very blurry. This is an inverse intensity image. But for each trajectory in principle gives us localizations with an accuracy of the order 40, 30 nanometers. So if we have enough trajectories, we can really use it to reconstitute a super resolution image, just as done in Palm or Storm super resolution microscopy. This is the result we get. Quite a bit of more information. And this is, if we compare that sort of, this is a cartoon based on EM structures, and this is if we compare that to our super resolution image. And you can really appreciate that close to the tip, the microtubules are very narrow. What is it? Of the order of 100 nanometers, much smaller than the diffraction limit. You also see they're wider here after the transition zone, and they're relatively narrow, 200 nanometers at the base. And I will skip the next slide. It takes me too much time because I would like to end the in vivo work to have some time for the in vitro work. And I hope I've shown you that the quantitative and single molecule view can really provide key insights into cellular processes. And the amazing thing is that you do not need any fancy fluorescence microscopy tool. You just need a very good camera and a very good laser and good optics. And wide field epifluorescence microscopy works. We've tried any other microscope and it doesn't work as well as our own. What we're looking forward to is to really connect IFT with cilium structure and cilium length. And we also want to connect information we get on IFT with the actual function of the cilium, and I can give you a big preview of that. We're now looking at one of the calcium channels involved in chemosensing called OCR2, which is a TRPV channel, and we're looking at how it is distributed over the cilium. So this is again one of these super resolution maps where you nicely see that it's embedded in the membrane. So here it's really, you see a hole. I'm shaking too much, but you see there is a hole. It's really localized on the side, so it's really embedded in the membrane. And it has a typical distribution, and we try to understand that. What you would expect, the cilia are not sticking out of the animal. Really only the tip is into contact with the outside world of the animal. So that's why you would expect these sensory molecules, proteins to be. So that makes sense. But we're trying to look into how it arrives where it should arrive. So we're recording many single molecule trajectories, and we get a very complicated picture, which we do not understand yet, but we're working hard on it. So what we see, this movie is not bright enough, we see that relatively bright blobs of this OCR2 protein arrive at the base of the cilium via the dendrite at very high velocity. So we think they're embedded in vesicles in the dendrite. They dock at the base of the cilium. And then we see really only active transport over the transition zone, this diffusion barrier, what I was talking about. Then we see sort of remarkable stuff. In the beginning of the cilium, we see a lot of diffusion, only limited amount of active transport, and we're really trying to find the right ways to sort of get numbers out, how much active and how much passive transport is going on. And in the tip again, we see much more active transport. And maybe next year, 2 years, we will have more. I hope to have more of a picture on that. So I promise you to also give you some insight in vitro experiments where we look at sort of this traffic jam behavior of motor proteins. In vitro has many disadvantages. Of course, you're simplifying everything. You only get out what you put in. Only the proteins are there that you have put in. But it, of course, gives you much more control than in cells or in whole animals. So that's why we try to look at that. And what we were really interested in, together with a group of Peter Schall from the University of Amsterdam, is to look at traffic jam behavior. They had been looking at models for real car traffic, and they were interested whether these sort of hold on the molecular level as well. Okay, let's join forces. So we modified our standard single molecule surface assay in the to make it happen, to make this work. So what we basically do is we use a relatively low number of labeled kinesin, and we add unlabeled kinesin at varying concentrations, and we look at how transport is affected. We still did this at pretty high concentration, so this is an example too high to allow single molecule tracking. Because they had some good ideas about using image correlation methods to get the motility parameters out of these kind of image sequences. How does this work? So basically what you do, you get subsequent images which are basically one-dimensional along a microtubule, and of course other dimension time. You can correlate them in both directions, time and position, and what you get is a parallelogram like this. Of course, the correlation disappears, and from this you can fit Gaussians, and from these Gaussians you can get two points of information, namely, of course, how the peak position changes with time, which gives you information on velocity, and also the area under the curve, that if you do the experiments well, that means that photobleaching doesn't play an important role, then that is related to the run length of the motor proteins. We tested this against single molecule tracking, and it works. And we applied it. So this is not the only thing, because the other thing what you really would want to know is that at a given concentration of motor proteins added to our assay, how large is the actual density of motors on the microtubule. And here you see sort of the effect. You see a couple of chimegraphs at different concentrations. Again, what is plotted here is the total concentration. So here we have 20 nanomolar of fluorescent proteins. Here we have 20 nanomolar of fluorescent and 20 nanomolar of non-fluorescent motors. And you really see enormous changes. So what we did is we used basically the ideas from fluorescence correlation spectroscopy, where you look at the fluctuations around the average intensity to determine locally the number of proteins, and we could do that. We studied two motor proteins, OSM3 and kinesin 1. Kinesin 1 is the standard kinesin that everybody has measured on, and This is the data we get. We can really get the motor density, motors per micrometer, as a function of the concentration of stuff we added. And then we can put stuff together. So forget about the red and blue lines for the moment. Just concentrate on the data. So in blue we see kinesin 1, and in red we see OSM 3. And just qualitatively looking at these graphs, there is a couple of things that are quite striking. First of all, that sort of the saturation density, so the point where you start deflecting from the flat line, is really hugely different for the two different motor proteins. And a second observation is that if you compare, for example, this red curve with this red curve, and it's the same for the kinesin 1, you see that something happens to the run length at much lower densities than that stuff happens at the velocity, which is also remarkable. Of course.
When the density here was related to particles that are where? I mean, density here of particles that were measured.
This is really the density on the microtubule.
On the microtubules, measured on the microtubules.
It's not the concentration added. We use this approach, this FCS-like approach, to really measure at a given concentration how many motors we have per micrometer.
So yeah, I have a question.
Yeah.
So what was the difference between the figure A and B?
So, sorry, this is run length.
Oh.
And this is velocity. I should have told
Okay.
you. Yeah. So we basically, we measure three things from our images. From the spatio-temporal correlation, we get velocity and run length. And from the sort of only the intensity correlation, temporal intensity correlation, we get the density. The density is plotted here on the x-axis, and run length and velocity there.
Sorry, but this is the density only of the labeled motors, right? Because you cannot know the total density.
It is using the information we get from the label, we calculate the total density. Yeah. Good point. So if you would assume a simple, or not simple, but a standard TASAP LK model with a binding rate, a detachment rate, and basically stuff can walk, and it just stuff piles up, then you would expect that velocity and run length would be affected at the same density. And that is not what we see. So we need to modify the TASAP LK model. And our collaborators, we discussed a long time about this, what could be happening here. And we have been trying different things to bring the theory closer to the experiments. So first of all, there is some evidence that the detachment rate for unconstrained motors that are just walking, is different than for constrained motors that are pausing because they cannot reach the binding site. So we added a difference there. For kinesin 1, I will show you, we really needed a longer interaction range than the 8 nanometers, which is the next binding sites. Otherwise we cannot understand our behavior. We have some ideas why that would be. And of course we're looking at microtubules, and microtubules have 13 protofilaments. You can of course argue for a very long time how many of them are accessible, are they independent? We assume here that they're independent and that we can just modify that. So we try to incorporate these things. So this is the data for OSM3 where we change different things. So again here it's so B and C are the same simulations, but this is velocity run length, and what we change here is we go from 1 lane to 3 lane to 6 lanes. This is a little bit of a, there is no dependency, you're just scaling things in the, what is it, the X direction. And here, this is the, what we changed is we kept it at 3 lanes, but we changed this difference between the detachment rate, constrained and unconstrained. So here it's equal, then we get the blue line, and then you see indeed that run length and velocity start getting affected at sort of the same densities. But to really get this difference, what we have observed, we really need to assume that the detachment rate for constrained motors is a lot higher, and 5 times that is what we found to be working. Okay, so now I show the data again, and now you're allowed to look at the lines, because these are in fact the best fits of our modified TASAP-LK model, and we could get very good description of the OSM3 without too many crazy things. 3 lanes were assumed. It's not super critical on that, whatever, but we can get a good description.
Do the motors switch lanes?
We do not actually know, but probably they do.
Okay, so I was asking if the motors switch lane. In the model, do you assume they don't?
No. So we really assume this is quite, because we do not have enough information to put anything wise in there. So we just assumed a 3-lane road with not with dotted lines, but with fixed lines where people are not, where the motors are not allowed to change. For kinesin 1, we really needed to stretch stuff a little bit, and the only way we can get a decent description is by assuming that the interaction range is not the next binding site, but that there is 60 nanometers, that they start feeding each other. And we scratched our head a very long time about that, because 60 nanometers, the motors, we assume them here to be 16 nanometers, so it's really a factor 5 larger. So what we think is going on is that, and there is some evidence for that from in vivo, where kinesin 1, specifically kinesin 1, only binds to bent microtubules. That's not published, but that's what some of my colleagues tell me all the time. And I recently heard a talk where somebody claimed that if you bind kinesin 1 to microtubules, that he could measure a small lengthening of the microtubule. So what we think is going on is that the kinesin modifies, by binding, modifies the microtubules lattice a little bit, and that could affect things. Hypothesis. Okay, conclusions about this in vitro work. Kinesin crowding effects can be effectively measured using turf microscopy and image correlation analysis. A modified TASAP-LK model can effectively describe the data, and we see remarkably different crowding behavior for kinesin 1 and OSM3. OSM3 can run effectively, also at rather high densities. And kinesin 1 is very, very sensitive to crowding. The interaction range is larger than its own length. Maybe it might deform microtubules. If we now take a step back again and look a little bit into the biology, then maybe this sort of makes sense. OSM3 really acts in on these IFT trains, where it walks in teams of the order of 30, 40, very closely packed together, while kinesin 1 is really thought to be much more of a lone transporter in exons. That might be the case. Okay, let me end here, but of course not before thanking the people who did the work. So the worm work has been done by Bram, Felix, Pierre, Jona, Seida and Jaap. in a collaboration that started with John Scuola in UC Davis. John retired now, but he still has a lot to say about this fascinating system. And the in vitro work is done, the experiments are done by Vandana together with Seda in my lab. And this was a collaboration with the lab of Peter Schall and Bernard Nienhuis in the University of Amsterdam, with Daniel doing all the image analysis and making the software for that, and Dimitri being in charge of the modeling. And these are the funding sources. And of course, I should not forget to thank all the lovely worms that left their life for science. Thank you for your attention.
Okay, so any questions?
Can you put the slides back up? Yeah, can you go back like 3 slides?
I hope I can.
Yeah, that's good. Yeah, thanks for this really nice talk. I really like it, and it's nice to see that you don't have to spend hundreds of thousands of dollars on your imaging equipment to get this kind of information. But yeah, I wanted to see the red and the blue curves that you told us not to look at. But
Yeah. Now here you were allowed to look.
Okay, well, I was having a bit of trouble seeing the effect that you were claiming, that there's a difference between run length and velocity, that there's a difference in where it turns over. I mean, there's a lot of noise. Probably there's not too many points on the right-hand end of the density. So, like, statistically, how much confidence do you actually have that that effect is actually there?
That's a good question. But I mean, yeah, but what I'm really pointing at is indeed the behavior. I think this one is, the red one is definitely not so clear, but I think it's more evident in the, how do you call it, in the Kinesin 1 data, where you really see that from, yeah, this sort of 10 particles, a little bit more than 10 particles per micrometer, it starts torping, and here it's really almost an order of magnitude larger. I mean, these are the what is it, standard errors of the mean. So you can argue what the statistical significance is, but.
Yeah, so I have a comment and a question. Since you have been able to look at on track with super resolution, have you used this information about many trajectories to actually find a sort of drift map to look at the velocity at each position, so you can correlate the position where you are, if you are in this transition zone or more? Yeah. So this is the first comment or question.
Yeah. So we have done some work on that, and again, it's a matter of statistics, but we can really see distributions of effects, and you really see that in certain regions we get a limited amount of active transport and much more diffusive transport. I can show you the data. I don't have it here, and that in other regions it's really taken over by the active transport component. Mm-hmm. Yes. Yeah.
And so, in a general comment, how generic or how stable is the mechanism that you have presented here across, for example, species, but more generally, for example, if we think about axonal or dendritic growth, we know that, I mean, a couple of years ago we looked at this, I mean, the analysis, that a dendrite is growing from, has a very different pattern of actually growing compared to axon, where axon is more like stable, coming to some kind of steady state, while the dendrite, there are more oscillation in the lengths. And I mean, it was not clear many years ago if this was due to the dynamics of these motors or something else, or actually we saw that it was the dynamic of the microtubule which could attach or detach to the tip.
Yeah.
And I think maybe with your technique you will have much more, I guess, to say about this. Do you have any comment on this question?
Not yet, but this is something we really want to look at. So it's clear that, for example, people have shown that certain components of, in particular IFTB, that they connect to EB proteins, so end-binding proteins. So that's something we really want to look into. If there is a specific affinity for some of these proteins for the tip of the microtubules. We have some preliminary data on the dynamics of the microtubules, and it looks like they're not as dynamic as other microtubules, but there is still a lot of dynamics going on. And we also have preliminary, less preliminary data where we perturb the system in different ways. We can do that by how to call it, relatively subtle ways, like putting in inhibitors of dynein. We can also do blunt methods, and blunt methods are much more fun, where we use a femtosecond laser to cut the dendrite that is connected to the cilium. And in both cases, we see that we see something that looks like a triggering of changes in the transport, which we understand as switching on the retrograde transport, activating the dynein to transport stuff back from distributed all over the cilium to the base of the cilium. And this is followed within a couple of seconds by a disassembly of the microtubules from the tip, but it stops sort of halfway where there is an actual axonal structure. You asked, so this is something that really has our attention, and we're diving into that. We're working hard on getting more insight into that. You were talking about the generality. I think the different transport mechanisms are quite different. I think one key difference, for example, compared to axonal and dendritic transport with IFT is that there it's almost entirely vesicles that are transported, organelles. In my simplistic mind, that should have enormous consequences for how motor proteins cooperate if they're attached to a membrane which is very floppy. Here it's really trains of proteins that have a very well-organized structure, so it might be much stiffer, and there might be much more mechanical interaction between the motors as a consequence of that. Second, cilia are quite different. So the basic structure and the basic machinery is very similar. If you compare Chlamydomonas, C. elegans, humans, and weird other bugs, but there are substantial differences. So for example, here we have the situation where we have these two kinesin motors working together. In most other cilia that have been studied so far, that is not the case, or in a different way the case. I think one of the issues is that these are very specialized cilia, and there is not from higher eukaryotic cilia, there is only very few that have been studied in detail. So these are just cancer cells, I don't know, but also the rods and cones, which are of course very specific cilia with a very specific structure. But for example, taste buds or those have not been studied in the detail as we have here. So I spoke with some other people about this. We think that there's still a lot to be discovered of this diversity and stuff. Yeah.
Okay, we have one last question. Thank you very much. Very nice talk. Do you think the motion patterns of these motor proteins, like their velocities, how fast they move, and the location they stop or start to move have some effects to the morphology of the cargoes, or what's the biological significance of their different species?
So what we think is that so in vivo it's not velocity that is actually regulated, it's really engagement with the cargo that is regulated, and I think that's what more people believe. So that really, if there is regulation mechanisms, it's really that it's detachment from a train and being picked up by another train. That's one thing. We clearly see that if we mess around with velocities by adding inhibitors or by making mutants, that has sometimes subtle, but it has effects on the morphology of the cilium. It really looks like, and in Chlamydomonas, people have spent careers on that. A major cargo of IFT is tubulin, and if there is not enough tubulin at the tip, that's the only place where it's incorporated. Then it immediately has effect on the length of the exoneme or the microtubules, and that's what we see here as well.
Okay, so let's thank Erwin again. And our next speaker will be Elena Koslover from UCSD. And she will talk about drift, wander or stick, disentangling modes of organelle motion in living cells.
All right, good morning everyone. Thank you for the chance to speak here today. And I want to continue this theme of intracellular transport to talk more about the different physical mechanisms, the interplay between them that move around organelles in eukaryotic cells. So just general background here, aside from the motor-driven directed motion that was discussed in the last talk, other mechanisms are available to drive organelles within the cell. There's diffusion, of course, just random wandering. There's also flow of cytoplasmic material that can pull organelles and other particles along with it. And on top of all of that, there is tethering or halting, often regulated in different biochemical ways, which, as we can see, can actually help distribute organelles in the cell. And so what I'm interested in primarily is how all of these different modes of motion combine to distribute organelles, how much the different modes contribute in different cases, and then what physical parameters can be leveraged to regulate organelle distribution. And I should mention here that my group is a theory group, so we use a lot of physical mathematical modeling coupled together with data that we get from collaborators. And ultimately this comes down to trying to extract the most information we can from data on how particles actually move in a cell, and also trying to map it to very simplistic, almost cartoonish physical models to get a sense of what's going on. All right, so I'll go pretty quickly through the introduction because I expect most of you have seen this before. The different modes of motion available for organelles in the cell. We'll start with diffusion or Brownian motion. The idea here is this is motion driven by some sort of uncorrelated fluctuations in the surrounding medium. And we could spend all day arguing about whether those are thermal fluctuations or active fluctuations. Either way, so long as they're uncorrelated, thermal-like fluctuations, you're going to get motion that is reminiscent of a random walk. So the base model for this is classic diffusion, white noise forces in a viscous fluid. And for such a random walk, you'll have a mean square displacement that scales linearly with time. And that scaling coefficient, diffusivity, is going to scale inversely with the particle size. So what this means is that the typical organelle diffusivity is much lower than the diffusivity of smaller particles. So for a typical organelle, diffusivity of about 0.01 micron squared per second, it would take about 2 hours to cover even a moderately sized 20 micron cell. Now there's additional complications one could layer on top of that. In particular, if you're inside a viscoelastic medium, your steps, even from just thermal fluctuations, are going to be negatively correlated. You're pushing through jello, essentially. Every time you take a step, you get pushed back the direction you came, in which case you're going to get subdiffusive motion. So mean square displacement scaling at some power and time that is less than linear. I apologize, the resolution here is not what I expected. This, for instance, is a fraction bar right here that's completely invisible. Hopefully this will not be too much of a problem. All right, so over and above diffusion, which of course is quite slow, you're not really capable of waiting 2 hours to get your organelles to where they need to go. Cells have developed this active motor-driven transport that involves motor proteins, like we just discussed, that burn energy to carry vesicles along cytoskeletal highways, like microtubules. For a sense of scale, this is the microtubule network in an animal cell. So these highways basically go everywhere throughout the cell. And these motors can have typical speeds on the order of about 1 micron per second, so allowing them to cover a cell in about 10 seconds. Now, additionally, a less well-characterized form of transport in eukaryotes is cytoplasmic flow. So it's been identified, discussed, characterized in certain specific cell types, for instance, in plant cells, where you get these slow cyclical flows around the outside of the cell that are driven by entrainment. So you get a lot of organelles being carried together in one direction by motors, pulling fluid along with them that entrains other particles. In other cell types, like this slime mold, Physarum, you can get much faster flows driven by peristaltic contractions. To some extent, you can see flows even in most crawling cells as you watch them deform. You have contraction of the myosin cytoskeleton in the back, pushing cytoplasm along as the cell crawls. So these flows can range from anywhere from nanometer per second to millimeter per second. And one of the main points I'm going to try to make is that they are a lot more common than previously identified. So they play an important role even in cell types where there's not a clear, well-organized form of flow, like inside these Physarum hyphae. All right, so I wanted to talk about two different stories today, two rather different examples of cases where different modes of transport contribute to spreading organelles around the cell. So one is going to be human neutrophils, white blood cells, and one is going to be fungal hyphae. And in both cases, we'll discuss a role for flow in helping to distribute the particles, and also the contribution of different modes of transport. So I'll start with these neutrophils, and this is work I did as a postdoc in Julie Theriault's lab at Stanford. Just a bit of background here. A neutrophil, it's a motile phagocytic white blood cell in the immune system. This is a rather classic movie of a neutrophil chasing down a bacterium and consuming it. And a key feature of these neutrophils is they are highly malleable. They really deform, change shape a lot as they move, and they have to be in order to squeeze through your tissues. They're also highly phagocytic, so they need to have rapid delivery of vesicles to and from their periphery in order to digest whatever it is that they engulf. Now, in the Theriault lab, I was working closely with an experimental graduate student who had a nice little setup where he had confined these neutrophil cells in 2 dimensions. So he has them squished flat under an agarose pad. There's a quick little anatomy of a neutrophil right here. At the front, there's a very dense actin network called the lamellipodium. The actin polymerizes, pulls that cell along, and it drags behind it a big bag of fluid of some sort, the cytoplasm, with a nucleus embedded in it. And so he would put these neutrophils in a uniform field of chemoattractant. They would randomly polarize and then start crawling in arbitrary directions. All right, so here's one such neutrophil crawling around. And if you focus on the texture of the cytoplasm, the various granules and organelles you see in there, you immediately get the impression that those are not just moving due to diffusion, nor does that really look like the sort of straight paths of motor-driven motion, such as we saw in the previous talk. Instead, the cytoplasm seems to be getting churned, squished around as the cell deforms. And so we wanted to address this question of what's governing the particle motion you see there. How much does flow contribute over the stochastic or diffusive motion? Right, so on the experimental side, what Caleb did was he labeled lysosomes, or acidified organelles in these cells. They're about half a micron across. They make convenient punctate tracer particles that we can track over time. We then track individual trajectories of the lysosome with time as the cell crawls, and then we can analyze those trajectories. In particular, we could look at the interparticle mean squared displacement here. So this is on a log-log scale, and you immediately see that there is a super diffusive scaling, more than linear with time, indicating some kind of precessive component to the motion. And I should emphasize here that this is mean squared displacement of interparticle distances. So the rigid body displacement of the cell, the fact that the whole thing is moving, is not contributing to these. Instead, it's the fact that different parts of the cell are moving to different extents that are helping drive the particles. All right, so two questions arose from this data. First of all, we wanted to get a handle on what we could say about the stochastic motion of the particles. So at short time scales, where the flow is presumably not going to play a big role, are they behaving diffusively? Are they embedded in something like a viscous medium? Or are we going to see subdiffusion? Are they moving in a gel? And then on longer time scales, what's driving the super diffusive motion? How much is flow really contributing to this? All right, so to focus first on those shorter time scales, right, the question is, there's some wiggles in those particle trajectories. Are those consistent with diffusion? Is the cytoplasm behaving as a viscous fluid or something more complicated? And to do this, we wanted to do a separation of timescales, focus on the short time behavior. And we developed a method for this called Brownian noise extracted with wavelets, because we had to call it something. I'll describe how it works on simulated data first. Right? So imagine you have some particle trajectories. They're diffusing, and on top of that, they're in some spatially varying flow field. And you want to know what is the diffusivity of those particles, and are they diffusive in the first place? Right, so you can't just look at the mean squared displacement, because this is a combination of two forms of motion, diffusion and drift. Instead, what we do is we smooth each trajectory using wavelets, so sliding window method, where you take a linear combination of data points in your window to estimate the velocity, integrate that over time, get a smooth trajectory. And then we subtract the smooth tracks from the original noisy data and focus on the residual noise that remains. So somewhere in that noise is information about the stochastic motion of the particles. Now, if you look at the mean squared displacement of that residual noise over time, short times it will increase, but once you get to a time scale comparable to your wavelet span, it's going to flatten out. No surprise there, because of course your noisy trajectories can't go arbitrarily far away from your smooth trajectories. All right, so how do we analyze these curves to characterize the stochastic motion? Well, the simplest thing to do would be to ask, what do you expect this to behave like if you have diffusion on top of a constant drift? And in fact, you can show that if you do this process of smoothing a trajectory with some wavelet for a particle that's diffusing and drifting in a constant direction, then your mean squared displacement of the residual noise will have a form that looks like this. It'll be the diffusion coefficient times some complicated but analytically known function, plus the localization error times some other analytically known function. Now, that form is simple enough, conveniently, that, again, fraction bars here and here, that you could rescale using those known functions to get your rescaled mean squared displacement that should behave linearly with a prefix as your diffusion coefficient and the localization here. All right, so those functions just depend on the shape of the wavelet you used for the smoothing. And since you know what wavelet you're using, this allows you to collapse your data from different wavelets onto a single universal line. You could then fit it to get the effective diffusion coefficient and localization error. So this sort of analysis would allow you to get rid of that slowly varying drift. This will work so long as the drift is varying slowly compared to the size of your wavelet, and to extract a measure of the diffusive noise. Yeah?
What are the units for the...
Right, so these guys are unitless. They've been rescaled according to...
In the previous slide, you gave an information about the diffusion coefficient, so yeah.
So, okay, these are simulations, so just
this is
arbitrary
simulation,
units.
okay.
Yes, this for now is simulations. I'll show them in real units in just a minute for real particles. Right, so the other convenient feature of this is if you have particles that are undergoing fractional Brownian motion. This is the usual model for particle motion in a viscoelastic medium, moving through a gel. Then you can do the exact same analysis, do the same rescaling with the same functions, and it will collapse not onto a universal line, but onto a universal power law. You could fit that, get your scaling exponent that characterizes the fractional Brownian motion. If that exponent is 1, these particles are behaving diffusively. If it's less than 1, you get fractional Brownian motion. All right, so using this analysis, not only do you get an estimate for the diffusivity, you also get some ability to diagnose whether they're exhibiting fractional Brownian motion or plane diffusion on short time scales. Okay, so now back to the real data with real units. If we take these trajectories of lysosomal particles in neutrophils, here they are in the cell frame of reference where you can still see some distinct drift, precessive motion of the trajectories. If we perform the analysis, look at the rescaled mean square displacement versus time, the trajectories nicely collapse onto a universal line. You have a typical fitted scaling coefficient of about 1. Here it is for many different cells histogrammed together. You get a broad range of diffusion coefficients with an average of about 0.004 microns squared per second. This is typical values for diffusivity in cytoplasm. So it appears that for these particles on short time scales, they are in fact behaving as if they're diffusing in a viscous environment. Okay, so the next question at hand then is what is driving the motion of these particles on longer scales? In particular, my cartoon picture is that I would think that the deformation of the cell is causing flow of the cytoplasm that's making these precessive, partially precessive tracks. To quantify that, we used a really simple model where we just treat the cytoplasm as a 2D slab of fluid. We trace the outline of the cell and track it over time. So as the cell crawls, we can see how its outline is changing shape. And we then use that as a velocity boundary condition to solve for what the fluid flow should be within the cytoplasm. So in some sense, it's a form of interpolation. Given what the boundary of the cell is doing, how much flow of the fluid inside the cell do you expect? Okay, one can then put simulated tracer particles into that flow and track their motion over time. All right, so encouragingly, if you look at the time-smooth trajectories of actual lysosomes and compare to these solutions of the fluid flow equations for what we expect the flow to be, you get reasonably good correlation, about 86% correlation on average between the predicted and measured motions. And if we look overall at that interparticle mean squared displacement over time, so these thin lines in the background are the results for individual cells. The overall average is this thick purple line here. And green is the results of simulations just based on the deformation of the cell boundary and the diffusion coefficient that we measured earlier. So you can rather nicely reproduce both the scaling and the overall magnitude of that interparticle mean squared displacement just by considering how much do you expect the particles to move given the cell is deforming. Now also, once you have those simulations in hand, you can ask questions about how much does the flow contribute over and above diffusion. So shown in yellow here is the results of the simulations where the particles are being driven by flow only. In black is diffusion only. You can see that above timescales of about 3 seconds or so, the flow starts to dominate. So these are very much biologically relevant times still, where flow is dominating above diffusion. All right, so what we've shown there is that the fluid flow seems to be enough to make the particles move more than they otherwise would move. Bigger mean square displacement, but does the cell necessarily care about that, right? Does it really matter if your mean square displacement goes up a bit? So we wanted to ask more directly whether this additional flow is helping in the mixing of the particles. And to do this, we made use of an even more abstract toy model to ask the rather general physical question. If you have a ball of fluid and you start deforming it, does the amount of flow you get actually affect the mixing of the particles? And what do your parameters have to be to make that the case? All right, so here's the general in silico sort of model we used. We assume a quasi-spherical system. So you start with either a 2-dimensional circle or a 3-dimensional ball, and then you're going to elongate it over time. The motivation here is if you look over a long time period at what these neutrophils do, they polarize, they crawl for a while in somewhat drifting directions, and then they depolarize, and then they start off in a new direction. So they are, in fact, kind of circular, and then they get longer and then circular again. And we know their typical size scale, about 10 microns. We know the period over which they remain polarized, about 2 minutes in between these turning points in their trajectories. Okay, so in our model, we take our ball, we assume the deformations are volume conserving and just have a single mode of elongation to them. But of course, that's not going to be enough, because if you take a ball of highly viscous fluid and you start deforming it back and forth along the same axis, you're going to immediately run into the so-called scallop theorem. If you have a highly viscous fluid, you make a reversible deformation to it, you're not going to go anywhere. You won't get any mixing. So, given that these cells do not, in fact, maintain a constant axis as they polarize and depolarize again, we'll have one more parameter that says, as the cell is polarized, what is the drift velocity for its polarization axis over the course of its deformation? So there's drift velocity gamma here. And in fact, you can see, I think this is for a two-dimensional system, if you just deform it along one axis, come back again, your color-coded particles at the start don't get mixed at all. If you allow the axis to wander as the cell deforms, you get this mixing, this Lagrangian chaos, as the particle distributions get kneaded together. All right, the one remaining parameter here is the diffusivity of the particles on top of this flow. Again, the diffusivity we know from previous measurements. All right, so this model is simple enough. It only has two dimensionless parameters. One is the dimensionless diffusivity. So this is the ratio of how long would it take the particles to cross the cell volume by diffusion versus the time period of deformation. It's essentially a Peclet number on the scale of the entire system. And then there's the dimensionless rotation rate of the axis. How far does it rotate? What angle does it cover over a single deformation period? All right, so we could take this model and ask the question of how much mixing do we get as a function of these two parameters. So for this, we'll need a metric of mixing. There's a lot of different ones people use. We decided to focus on a simple one the cells might actually care about. If you start with a bunch of particles near the center of your domain, you could simply ask, what is the first passage time for particles to get to the domain boundary? So how quickly do particles go from the center to the border of the cell? All right, so this is the fraction absorbed at the boundary plotted versus time. In black is the solution for just diffusion within a spherical shell. And as you start increasing the drift of your axis, so increasing the mixing due to this flow, you increase the rate at which the particles reach the boundary. So to characterize that by just a single number, you could simply fit this to the functional form for diffusive transport within a sphere with some different effective diffusivity. So essentially you're asking, given how quickly the particles are hitting the boundary, how diffusive would they have to be to do that by diffusion alone? Just a convenient way to get a single number from these curves. All right, so if we look at that effective diffusivity versus the axis rotation rate, some different particle diffusivities here. We see that as the axis rotation rate gets large, you do in fact get an enhancement in particle mixing for parameters relevant to lysosomes and neutrophils. Again, we get those from just looking at how the neutrophils deform and our measurement of diffusivity. We are right around here. So diffusivity about 0.007, gamma of around 1, something like 30 or 40% acceleration in how quickly these particles hit the boundary. All right, so basically what this is telling us is that the rate at which neutrophils deform, the diffusivity of organelles within them combine to indicate that the flow of the cytoplasm is enough to accelerate substantially the rate at which organelles can reach the periphery of the cell. Yeah?
I'm curious about how the effect, the enhancement of diffusion is not as big when the diffusion coefficient itself is much larger. Is that because things are already reaching the boundary in one cycle, and so you don't do any better by squashing them?
Mm-hmm.
Okay.
Yes, so if the dimensionless diffusivity is 1 or close to 1, that means in the time it takes for the cell to deform and the fluid flow to actually get set up, the particles have already reached the boundary of the cell. Yeah. So keeping in mind diffusivity is also dependent on the size of the particle. This does mean that protein mixing is not going to be affected by these flows, proteins being an order of magnitude smaller than organelles. Yes?
Maybe you've said this before, but is there an optimal amount of mixing here, kind of like reminiscent of species diversity and mixing in ecosystems, or do you just want maximal amount of mixing? Or?
Yeah, that is a fascinating question, because I've just declared that mixing is something useful for the cell to deliver its organelles. That's not at all obvious. It might be much more useful for the cell to position the organelles where they need to be and then fix them there. My suspicion, and this is entirely not backed up by any data, is that in general there's a lot of biochemical mechanisms to fix organelles where they want to be once they get there, and so the cell is just limited by getting them there to begin with. But I wouldn't be surprised if there is reasons to avoid overly fast organelle transport, because you need enough time for those biochemical mechanisms to take play.
But in the system, they're continuously moving.
They're not getting fixed. These particular organelles, yes. There's plenty of other organelles we're not tracking. Because these particular organelles, their role in the cell is to go back and forth from the cell periphery to the nucleus. These are lysosomes, so they merge with things coming from the cell periphery, help digest them, go back to the nucleus, come out again. So I don't think these ones need to be in a particular region of the cell. Other organelles do. Right, so maybe I should say more specifically for lysosomes, I don't see any reason having them move arbitrarily fast would be a bad thing. Okay. All right, so at this point, I want to switch over to another model system, in particular the motion of peroxisomes in fungal hyphae. So this is work it's been done in my group in collaboration with the Rick Peterson lab in cell and molecular medicine at UCSD. Different type of organelles, different cell type, different geometry, but some analogous effects at play. So in particular, the system here are these fungal hyphae tips. They're only a couple microns across, something like 40 microns long. And the Rick Peterson group tracks peroxisomes, 200 nanometer organelles within these hyphae. So this is a particularly convenient system for modeling transport because, like the flagella we heard about earlier, they're essentially one-dimensional. Makes all of your models a lot easier. Okay, now from published data in different fungal hyphae, it's been shown that there are a number of transport modes available to these peroxisomes. First of all, there's passive diffusion. So if you basically knock out active transport by turning off kinesin, or rather turning off the hook proteins that link organelles to kinesin, you get a diffusivity of about 0.005 microns squared per second for these organelles. On the other hand, if you look at wild-type cells and focus on parts of the trajectory where the organelles are moving as essentially a random walk behavior, they at least in published data, exhibit diffusive scaling with a diffusivity of about 0.015 microns squared per second, so about 3 times faster than when you turned off active transport. So this has been used to imply that the active motion of various organelles is somehow helping to churn the system to increase the diffusivity to this active diffusion level. And I want to speculate here that the possible source of this is in fact hydrodynamic entrainment, that if you have different organelles passing back and forth, they are churning the cytoplasm, increasing the random motion of the peroxisomes. But over and on top of these sources of diffusion, peroxisomes are also capable of precessive motion. In particular, they use a non-canonical form of transport called hitchhiking, where instead of directly coupling to molecular motors, they sit around, they wait for an endosome to pass by. The endosome is in fact being carried by molecular motors, and it will transiently link to a peroxisome and drag it for some period of time. So these hitchhiking events are quite fast, about 2 microns per second on average. They are very long, can go up around 6 micrometers, typical run length, but they're very rare. So only about 2% of peroxisomes are engaged in these precessive runs at any given time. All right, and so the question we want to ask is how much do these different transport modes contribute? How much does these fast but rare hitchhiking events actually help distribute the peroxisomes throughout the cell? All right, so I'll start with this issue of active diffusion and how much additional diffusive motion do you expect from entrainment due to passing organelles? So the toy model we used for this was just considering the hypha as a no-slip cylinder, imagining an organelle passing through the center of that hypha. So this is your endosome walking by, and then you can solve for what you expect the flow field to be within that cylinder. Conveniently enough, there's an analytical solution available for flow due to a Stokes slit in a no-slip cylinder. And the main feature of that solution is that your flow field decays exponentially with axial distance from your Stokes slit, which means that if you are an entrained particle caught up in this flow, then you're going to lose sight of that passing endosome over a finite time period. And so that means your entrained particle goes on a finite trajectory every time an endosome passes by it. And of course, it'll go further if it happens to be near the center of the axis. Further away towards the boundary, you'd actually expect it to move backwards. This is just conservation of mass to fill in the fluid behind the endosome. If you look at those run lengths and assume a uniformly distributed radial position of the peroxisomes, you get a typical run length of about 75 nanometers, so comparable in size to the peroxisomes themselves, meaning every time an endosome or other organelle passes by, the peroxisome jiggles around or moves in one direction with a run length of about 75 nanometers. We know how often endosomes pass by, roughly 1 per second. And so we can put those together to get an effective diffusion constant, just treating this as a random walk motion, where once every second you take a random step of 75 nanometers. You get a typical diffusivity of about 0.01 microns squared per second. So very much within the order of magnitude that would explain the difference between the overall diffusivity in wild-type cells and the diffusivity you get once you turn off active transport. So it appears that even just looking at endosomes alone, the amount of entrainment you would expect to get is enough to account for this enhanced active diffusion. All right, so now we can ask, how does this couple together with hitchhiking? So if you have organelles that are capable of diffusing, actively or not, and going on precessive runs, how much do those two modes of motion contribute to their dispersion? And so for this, we use the simplest possible model. We call it a halting creeper model, where you have an organelle that's in one of two states. It's either diffusing or it's walking in one direction or another. And you have a characteristic rate of switching between the two states. Now, the nice thing about such a model is it is, in fact, analytically tractable. That is, at least in Fourier Laplace transform space and time, you could calculate the full distribution of the particle. Meaning if I tell you it started diffusive at time 0 and it's diffusive at time t, I can tell you exactly the distribution of where it is in space. And you do this through a convolution of the propagators for diffusive and walking motions. All right, so this allows us to calculate things like the range of the particle over time. Range being how much of the domain of the line has the particle covered over a certain time period. Not too surprisingly, the range shows some scaling transitions, where at short times, the range is just determined by the diffusive motion of the particles. F here is the fraction of particles in the walking state. So 1 minus F is the fraction of the diffusive state. At some time point, it switches over to a more ballistic-like scaling as the walking of the particles kick in. There's some characteristic length scale, x star here, where the precessive motion starts to dominate the behavior of the particles. And if you plug in some reasonable parameters for peroxisomes, this length scale is about 0.8 microns. So above about 0.8 microns, the precessive motion starts to dominate. At further time scales, you get a transition again as the particles have time to take multiple runs back and forth in different directions, and so their behavior looks diffusive again with some larger effective diffusivity. Okay, so the nice thing about the range is it allows us to calculate certain search properties of the particles. In particular, you could ask the question, if you have a uniform field of particles at a certain density, and you have some target that appears in the cell. So peroxisomes, for instance, are responsible for digesting harmful metabolic byproducts. So let's say one such byproduct appears somewhere, how long until a peroxisome happens to reach it? This hitting time can be expressed in terms of the range, and it's plotted here for two different parameters of the model. Here's the diffusivity, here's the fraction of particles walking. The black line indicates the transition between two regimes. If you're above that line, it's the diffusivity that primarily determines how quickly you reach your target. Below that line, it's the precessive walks that determine how quickly you reach the target. If we plug in parameters for peroxisomes and fungal hyphae, we actually find that we're right around that transition region. So both diffusivity and active walk seem to be contributing to how quickly they move on the length scale of separation between multiple peroxisomes. So again, about 0.8 microns. All right, now more recently, we started to get data from the Reck-Peterson group about peroxisome motion in a slightly different fungal system, Aspergillus nigilans. And one of the first things they asked us is, they think the particles might be tethered to microtubules. We'll get to in a minute why they think that's the case. But this motivates complicating the model a little bit to consider what happens if you do have a tethering state for the particles. So the idea here is you're going to get two different effects from tethering them to the microtubules. On the one hand, when they're tethered, they're not diffusing, so you don't spread as fast. On the other hand, if you have not very dense microtubules in a wide hypha, then tethering will make you spend more time near the microtubule, which means you're more likely to get picked up for a processive walk. So those two effects would, the interplay between those might determine whether tethering is helpful or not. So we can introduce tethering into the model, do the same sort of calculations, and find that tethering is going to aid transport. It's going to increase the range of the particle over time above some critical length scale. And you can analytically find that critical length scale. Depends on the relative size of the region within which these particles can bind to the microtubule for walking or tethering, and the fraction of particles walking, and the velocity versus the diffusivity of the particles. And again, for peroxisomes and fungal hyphae, the critical length is about 0.8 microns. So that means that tethering is neither going to help nor hurt how quickly these peroxisomes move to find a target, given how closely spaced they are, but it is going to help, it is going to greatly help establish the distribution of the peroxisomes to begin with over the scale of the entire hypha. Okay, so the last part I want to talk about is some analysis of this new data coming out of the Peterson lab, where they have 3D lattice light sheet microscopy. They label both peroxisomes and endosomes in their hypha. And the first thing we see is pink here is the peroxisomes. When they're not running, they actually exhibit very little movement. So this is what led them to speculate that maybe they're being tethered in the first place. Right, so we wanted to analyze these passive motions of the particles to see if their behavior is consistent with tethering, or maybe they're just diffusing with a very low diffusivity. All right, so to do this, we need to take particle trajectories, break them up into pieces where we identify the pieces of processive motion versus not processive motion. This is rather straightforward. Methods for this have been published before. You basically do your sliding wavelet again, and you use a universal thresholding technique to see where the velocities are much bigger than you would expect from noise alone. Label that as the processively moving components, and then focus on the pieces of non-processive motion that remain. Okay, so the first thing that comes out of that is if you look at those passive peroxisome trajectories in anidulous fungi, then you find that they are severely restricted compared to what you would expect from the published data on peroxisome motion. So this black line here is diffusive motion with the published diffusivity in a different species. To be fully honest, I have no idea yet whether it's the species that matters or one of these hyphae in two different labs are healthier than other hyphae or what's going on. So we haven't quite figured out where the difference lies right now. But in the data we have available to us, these trajectories are much slower and they're pretty clearly subdiffusively scaling. In fact, if you look at the mean squared displacement over time, they scale as t to the 0.75 power, 3/4 power, with some small localization error that's about what you would expect from these sorts of fluorescence microscopy measurements. Now, this scaling is rather evocative because the scaling with time to the 3/4 power is exactly what you would expect for the dynamics of a semi-flexible polymer. So you have a worm-like chain, a semi-flexible polymer, wiggling around in viscous solution. You would, in fact, expect to see this exact scaling. All right, but that's just mean squared displacement. If you look at some other metrics of the motion, we could consider the velocity autocorrelation function. So this has been used before to characterize particle motions. The idea here is you define velocity over some time scale, delta. Here, delta is 1, 2, 3, 4, 5 steps, I think. And then you look at the velocity measurement at one point, and then at another point later in time, you dot them together, and then you take the average, time averaged, ensemble averaged. You get a velocity autocorrelation. So it's plotted here versus time, normalized by what you would expect at 0 time. And the different colors here correspond to different delta values. So the velocity being defined on different time scales. So this right here is experimental measurements for peroxisome trajectories. To interpret that, we need to think about how these velocity autocorrelations would look like for some different physical models. So here's some simple models shown here. If you have diffusion only, as soon as your velocity windows are not overlapping, you're going to get no correlation at all. So these velocity autocorrelation functions go to 0 and then stay at 0. If you have some amount of localization error in your data, you will get some negative peaks in the velocity autocorrelation function, but those peaks will disappear as you measure your velocity over larger and larger separations. And if you have fractional Brownian motion with a particular scaling, you will get negative peaks that just stay constant over time. Now, the experimental data honestly does not really look like any of these. In fact, it starts low, it comes up high, and seems to asymptote. We can try overlaying it with fractional Brownian motion with the correct scaling. It really does not match. However, what we can do is take the model we had in the back of our mind all along, that these particles are tethered to a microtubule, and then ask, what do we expect for the velocity autocorrelation of a particle attached to a semi-flexible chain? So for now, these are just Brownian dynamic simulations. We're still working on the analytics. And you attach a particle to your chain, you run it over time, you get trajectories. All right, so there's some basic parameters that go into this. You need a radius for your peroxisome. We used about 100 nanometers. You need a diffusivity. We use the same diffusivity from the published data in that other fungal system. You need some length of the microtubules. The results are not actually very sensitive to this length. And you need a persistence length. So how flexible are the microtubules? We use the value that is consistent with what's previously been measured for bending of microtubules in vivo, inside living cells. Okay, so I won't pretend that there's no fitting parameters here, because of course we could have taken different choices of some of these parameter values. But nonetheless, these are parameters that are quite reasonable from what's previously been expected from different measurements. And if we run the simulation with these parameters, we actually get really nice matchup to what the peroxisome motion looks like, both in terms of the velocity autocorrelation, black here is the simulations, colored is the peroxisomes, and in terms of the mean square displacement. All right, so this would seem to imply that, yeah?
So, yeah, this is a question like for possible model. So your microtubule is basically taut on the scales you're looking at.
Basically what?
Like taut, like straight, like small.
Yes,
it's linear
it does not wiggle too much because it is quite stiff.
Okay,
yeah.
So maybe, I mean, this would look like you'd have exponential correlations then. So you could, I mean, you could basically do a linear decomposition of the modes of the motion. So you should get like a sum of exponentials, basically. This could maybe explain what you say.
Except, I mean, the way polymer dynamics works, right, is over time, more and more of the polymer starts relaxing, which is why you get these power law scalings to the relaxation. You have an infinite number of modes of motion in some sense.
Yeah, exactly.
Right.
But, well, maybe we can discuss later. Maybe, yeah.
No, I mean, yes, this can be done analytically, but we stumbled across this 2 weeks ago, so we just haven't finished it yet. Yeah. There's also some issues in that, you know, you'd have to deal with this larger particles here, and it's not clear whether you are in a transition regime where the boundary conditions really matter, or if it's all, you can do this with an infinite chain or not. But yes, this is definitely the direction we're going in right now, yeah.
I didn't understand the model. You have a sphere tethered to a material point on the worm-like chain?
Yes.
So it's not diffusing along the arc length?
No.
Okay, and then to do the 2-point, the velocity correlation functions, you'd have two spheres. Is that the idea? That?
So you look at this sphere over time in your simulations. You get essentially a trajectory, and then your velocity
is... autocorrelated.
It's autocorrelated, yes.
Okay,
yeah.
Got it, thank you.
Okay, yes, so obviously this is all still rather preliminary, but it is at least interesting that you could start with parameters from previously published motion of the peroxisomes. You could say, all right, now what if the peroxisomes are tethered, and that would exactly reproduce what we see in the actually measured peroxisome motions in this system. All right, so I'll wrap up here. Emphasizing maybe, first of all, this role of cytoplasmic flow, disorganized cytoplasmic flow, in one case arising from cell deformation, in the other potentially from entrainment due to passing organelles, both helping to contribute to the mixing of particles. And then also the fact that these cells are, in fact, leveraging multimodal motion, some combination of flow and diffusion, or flow plus motor-driven transport to enhance how they spread their organelles. And of course, I want to thank the people who did a lot of this work. My grad student, Saurabh Mogre, did a lot of the modeling for the peroxisomes. Caleb supplied all the experimental data on the neutrophils, and Cassandra supplied the experimental data on the peroxisomal motions. Thank you for listening.
Could you just say a few more words about the definition of your halting creeper model?
Yeah, it's literally the simplest thing you could think of. 2 states, or I guess technically 3 states: diffusing, walking left, walking right, constant rate of switching from walking to diffusion, constant rate of switching from diffusion to walking. A Poissonian process for both.
There's a diffusional walking that wins at the long time scales.
At long time scales, walking will win, yes.
Right.
Okay, technically not for all parameter regimes. There's a maximal Peclet number for the system, which is, if you look at the Peclet number on the time scale of a single walk before it turns around. If that Peclet number is bigger than 1, then walking is important at long lengths. But for these systems in cells, that Peclet number is always way bigger than 1. So at long time scales, walking will win, yeah.
So can you, thank you, very nice talk. Can you tell us something about what your goal in the analyses presented so far are, especially from the viewpoint of us to be, being able to estimate the extent of sort of overfitting or choosing the model that best fits the data, and the number of free parameters and so on, and and the whole picture of what it is we're trying to get out of this.
Right. So I guess to answer that in a very general sense first, my goal is always to start with the simplest possible thing I could think of that encompasses the cartoon picture people have of their system in their minds. Right. So when people talk about transport of an organelle and it's bidirectional, so we know it can go in either direction, and we know that there's some pauses in between there. The simplest thing I can think of is that you have 2 states and there's a constant rate between them. And then I want to go from there and say, what are the consequences of that? Does that match the different experimental data, and what does that mean for the cell? And then where it doesn't match, we start adding on other complications, like where these particles seem to be tethered, so let's add tethering to the model. Similarly for the neutrophils, you look at those cells crawling, the first thing you see is this is not diffusive motion, and it does not look like motor-driven motion. All right, the cartoon picture in your mind is something's being squished and turned around. So what's the simplest thing we can do there? Okay, take a blob of fluid. How much motion do you expect to see?
But even there, some of those organelles are going to be tethered to microtubules, which are also feeling forces. Right?
Some of them are, yes, which I would say is like it's an extra level of complication, right? So this seems to indicate, and actually, I didn't show. Can I flip back to the slides? Okay, just for fun. When you take that really simple system where you just have a blob of fluid and it's moving around, and you say, how much motion do you expect? No fitting parameters, right? This is just what do the actual cells do. You totally reproduce the motion of the scaling, the magnitude of the motion of these particles. So yes, some of them may be tethered to microtubules, but you don't need that to reproduce what the particles seem to be doing. And you could add that on and say, okay, what extra stuff do we get from that?
But what's the conclusion that we're to draw from this? What you're saying is there's a mixture of 3 different behaviors, and
so the conclusion for these particular organelles is that flow is important compared to diffusion, and you don't whatever active transport there is, and honestly just by eye you don't see a lot of directed transport of these particles, is not necessary to explain their motion in the statistical average.
I had a question about the relation between the early models you showed in your talk and the observations of lots of published articles, whether or not means they're correct, of subdiffusive behavior of smaller objects, molecular species, going all the way up to macromolecules, like the 100 nanometer sized things in the papers by Golding and Cox, where they did a very good job in the absence of active motion of showing subdiffusion with no moving boundaries per se, because those were bacterial cells without any flexibility of their cell walls and things like that. So, but in your models, of course, you're not seeing any subdiffusive motion in those.
For these organelles, their motion seems to be consistent with this diffusive-like motion. And I mean, I think there's almost a cottage industry at this point for different cell types and different particles deciding whether they're subdiffusive or diffusive motions. I think a lot of it is cell type dependent. Certainly in bacterial cells, I think the evidence is very strong that everything is much more gel-like than in eukaryotic cytoplasm. And there's people who argue in a eukaryotic cytoplasm, also on that 100 nanometer scale, it is gel-like. But it's not clear whether that's general to all cell types, or whether these cells, given how easily deforming they are perhaps more fluid than other cells. So I think it's one of those things where for every new cell you study, you have to re-ask that same question of, is it a fluid or not?
Yeah, and I'll just add a comment that in the active matter field, not biological necessarily, one of the popular models gets rid of translational diffusion completely and puts persistent motion plus rotational diffusion only. They find in that, this is called active Brownian particles. They can zero the translational diffusion or make it finite. They see no difference. All they need is rotational diffusion to be finite, plus some scale of persistent motion. That gives rise to a very rich phase diagram of different behaviors. So I wonder if...
That's an interesting notion. It's not off the top of my head clear to me physically what would account for that kind of behavior with these organelles. Because I guess the idea is like swimming bacteria, where they keep going forward, but what forward is randomly changes over time.
Yeah, that's one application, of course. Yeah.
Right, but then these organelles, if they are moving processively, if motors are carrying them, then no, they don't have rotational diffusion.
I'm not saying it applies to motion along tracks or in the presence of
no, I mean, it's a fascinating set of
long
models.
linear objects, but
Yeah.
I just made a comment, thanks.
Okay, I think we need to make a break, but feel free to just go and talk to Lena, and let's thank her again. And we'll start again at 11:30. Our next speaker will be Martin Evans from University of Edinburgh. He'll talk about antimicrobial resistance, modeling the efficacy of antibiotic treatment.
Is that better? It's there. Oh, I see. I'm getting confused by the picture of me. So anyway, thanks for the organisers for inviting me, and I think there's people from many different backgrounds here. So I should just say, first of all, I'm in a physics department, but I'm fortunate enough to be in a sort of broad group. It's soft matter, statistical and biological physics. So I can interact with people who are doing sort of biology full time. And this is just a nice picture of Edinburgh to inspire people to come, I guess. No. In the middle. Yeah. Okay, so the title of my talk is basically a model of antibiotic action on bacterial growth. And I think it's simplest if I tell you, why I'm interested in what I want to tell you. So I've been doing theoretical physics for, doesn't bear thinking about 25 years or so. And this is the first time we wrote a paper where we had a theory and experimental data, and they agreed. It's actually, one other time I had a paper with theory and experimental data, but the agreement was to within an order of magnitude. And here we actually get data collapse, which is what one likes as physicists, and I'll explain what that means. So what I should say is the people involved, Philip Grölich, he was a postdoc in Edinburgh. He's now got a position in mathematical biology in Southampton. Interestingly, the sort of hero of this paper is Matt Scott, and he's in the applied mathematics department. So it's sort of very interesting times when the experimentalist, because he did all the experiments, is the mathematician. So that, to my, that's sort of the world gone slightly upside down, but it's worked very well here. And Rosalind Allen is my colleague in Edinburgh, and she started off in theoretical chemistry, did PhD in theoretical physics, and now is a biologist. So what I'm going to talk about, I'll try and keep it broad. So the first part is hopefully very broad, and it may not be news, hopefully won't be news to you. So it's just really about antibiotics. And then the main part of the talk is to describe a simple model of dynamics in the bacterial cell. So a simple model of antibiotic action on bacteria. And then we compare with experiments. Actually, okay, those two are in opposite order. But that's the main thing, is the simple model in comparison with the experiments. That's it. So anyway, I grew up in Scotland, and so the story of antibiotics, we got taught at school that Alexander Fleming was a sort of a paradigm of Scottish scientific endeavour. Just when I was preparing this talk, I actually Wikipedia'd him. So indeed he was Scottish, but his family moved to England when he was 13 or something like that. And in fact he did his work in London, the work that he's famous for. So we all got taught the story of Alexander Fleming. He was sort of a careless experimentalist, careless around the lab, left things around, and then a story as he went off for, it was actually a long weekend, so holiday weekend, and he kind of left some things around. So he left some Petri dishes with agar gel, and he left the window open. And when he came back, the agar gel of course had bacteria growing on it, but there were a few spots where spores had come in through the window where the bacteria had been killed. And he recognized, this is a way of killing bacteria. And the rest is history. So, I mean, I guess, is that story sort of, you get taught that in school and other places? Yeah, okay. So apparently he was also, as well as being careless around the lab, he wasn't a very good communicator. So it took a while for him to sort of really get the idea going that this was this was a big thing, a big discovery. But nevertheless, by the 1940s, penicillin was being mass-produced, American drugs industry, and indeed it was used towards the end of the Second World War. And it's been a major game changer, and one way of looking at this is the leading causes of death, let's say around about a century ago, were pneumonia, tuberculosis, diarrhea, and, well, at least for bacterial pneumonia, all of these can be cured by antibiotics. So the world has changed, and now we have other things to worry about that are the main causes of death. But more than that, it's a sort of part of day-to-day life in hospital, the use of antibiotics to clean the wards, etc., and make sure everything works. So without antibiotics, they would be in big trouble. Now, why am I getting this wrong? That's it. The big problem is the rise in antibiotic resistance. And this was first noted by Alexander Fleming himself, that he noticed that things soon became resistant to penicillin, even over the few years that he was working. And it's actually not surprising, because, although he discovered antibiotics, they've been around for millions of years. So it's natural that bacteria had developed strategies to cope with something that kills them, basically. So that it's natural that they had worked out ways of evolving. And indeed, they are evolving. And so we've got a rise of antibiotic resistance, strains that are resistant to various antibiotics, and the number of new antibiotics is not going up commensurately. So there's clearly a problem ahead. One suggestion is hey, why don't we just back off and stop using so many antibiotics? So because there's a cost for the bacteria to be resistant, if we stop using antibiotics, maybe they'll become unresistant. Unfortunately, that's not going to work. The timescales are far too long. So something has to give here. So I'm not claiming that we can solve this problem yet, but what we need is at least a good models for how antibiotics work. Because surprisingly, from, if you like, a physicist or a systems biology viewpoint, a lot of the work is known about what an antibiotic does, like where it binds and what action it has. But the system effect of the antibiotic is not so well studied. And that's what we wanted to have a look at, sort of the collective effect of the antibiotic on the bacterial population. So that's what I'm going to talk about. So first of all, just comment briefly here, because I'm not an expert. Antibiotics work by targeting the difference between bacterial and human cells. And the ones I'm going to focus on are called ribosome targeting antibiotics. So basically, ribosomes translate the RNA and lead to productions of proteins. So if we can stop that process, then it stops protein synthesis and it stops the bacterial cells growing. That's how they work. So we'll be focusing on those. And then there's a rough classification that's not globally accepted, but nevertheless it's there in two different kinds of antibiotic action. Either bacteriostatic, which means it slows down the bacterial population growing and basically stops it growing, or more extreme is bactericidal, where it actually kills off the population. And we'll come back to this. We can explain this classification in a simple model in broad terms. Okay, so from a physicist's point of view, bacteria is very nice. It's very simple. It doesn't have any organelles, so we can just, or cell nucleus. So we can just very, very crudely, and in what we call a coarse-grained way, think of it as a sort of tin of soup, where it's a sort of homogeneous mixture, and we just kind of concentrate on the ribosomes. So as I said, the ribosomes lead to proteins and lead to cell growth, and then we have some ribosome synthesis. And then basically the antibiotic is going to inflow and break up this process, and we want to sort of make a model for that system. So what one measures experimentally is the doubling time for the bacteria, the time for the population to double under certain imposed conditions. Now in physics we prefer to think about growth rates, but that you can easily relate a growth rate lambda to a doubling time TD. It's, okay, if you do the maths, lambda is log 2 over TD. Right? So I will talk about growth rates, and the 0 there is meant to be the base growth rate. So we fix the medium and we fix the bacteria, and there's going to be a growth rate for the population in the absence of antibiotic. Then we apply an antibiotic, and that growth rate, if the antibiotic works, should decrease. So then we get a function called growth inhibition curve, which we can determine experimentally, lambda as a function of A, the antibiotic concentration. And the IC50 is the antibiotic dose which halves the growth rate. So it's the value of A at which lambda goes to lambda naught over 2. And then the standard wisdom is that, okay, if a bacterial population is growing too fast, then somehow it's like an army that overextends itself and is vulnerable to attack. So the received wisdom is that it's easier to kill off or attack a bacterial population that's growing fast than one that's growing slow. So the received wisdom is that the slower growing bacteria are more resilient and so should have the higher IC50. And we'll come back to that in a moment. Okay, so these are the experiments by Matt Scott. So basically he's growing bacteria under different nutrient conditions. So the bacteria is fixed, it's some strain of E. coli, the medium is fixed, but then he can add in glucose, for example, or, no. How did I jump? I'm giving it all away. So we can add in glucose and amino acids and nucleotides, and this is going to, these are sort of yummy stuff for the bacteria, so it'll increase the base growth rate. Then glycerol isn't quite so yummy as glucose, so there's a lower growth rate there, and he can get 6 values of a base growth rate, so that's without any antibiotics. So then he can fix those media and then add antibiotic to get 6 different growth inhibition curves. And he did it for 4 different standard antibiotics. Chloramphenicol, tetracycline, streptomycin, kanamycin, and 2 of these are sort of generally classified as bacteriostatic and 2 are bactericidal. So what happens, this is for the bacteriostatic ones. So these are these growth inhibition curves, but remember here it's normalized, so that is lambda over lambda naught. So they, as they should do, they go down with A. Then the interesting thing to think about is where it goes when we increasing the base growth rate lambda naught. So this is increasing lambda naught. The arrow goes this way. And as we see, as that increases, we actually decrease the IC50. Right? Remember, IC50 is the value of the concentration at which this goes to a half. So this is decreasing with increasing lambda naught. So that's plotted here. So this might look like it's going all over the place, but one should separate glycerol from glucose. So this is for glycerol, it goes down, and for glucose, it also goes down. So that fits in with the received wisdom. There, for chloramphenicol, we get a similar picture. Here it's not quite so clear. If you look at the chloramphenicol, Femincol, the IC50 goes down, and then it's not quite so clear. And we'll come back to that point later. But generally, the growth inhibition curves are smooth curves, and the IC50 goes down with lambda naught. So now we move on to the results for the bactericidal antibiotics. And things look very different. So first of all, these are not so smooth. They have a threshold when they're constant, and then they sharply decrease at roughly some threshold value. Right? So it's more like a threshold curve. Then if we increase the base growth rate, we're actually going this way. So if you plot the IC50 against the base or the drug-free growth rate, it actually increases, which is contrary to the received wisdom. And again, we get the same behavior here for kanamycin. The threshold behavior in the drug inhibition curve and the increase, okay, let's say glucose, we get an increase of IC50 with base growth rate. Okay, so that's the experimental result. So just to summarize, just to say what I've done again, for bacteriostatic, the faster growing bacteria are more susceptible, and we get nice smooth growth inhibition curves which decrease slowly or smoothly with the antibiotic concentration. For bactericidal, can and strep, the faster growing bacteria are actually less susceptible, and the growth inhibition curve shows this sharp decrease. Above a certain value, above a certain concentration, the antibiotic starts to kick in. Yeah.
So just for clarification, all these antibiotics are targeting the ribosome, right?
Yes, they're all ribosome targeting in different ways. They target different things along the RNA, but basically they all inhibit the ribosome. Okay, so that's the results that Matt carefully did. We wanted to try and explain them, and explain in particular why we got this different behavior. So we kind of made the simplest model. Again, going back to this physicist picture of a kind of tin of soup. I have to make a couple of corrections here to this picture. This has a couple of things missing. But the important thing is that we want to characterize the inside of the bacteria. This is the bacterial cell. We want to characterize it by three concentrations. The concentration of antibiotic, which is A. The concentration of what we call inactive ribosomes. So these are ribosomes where an antibiotic has bound to it and has knocked it out. And I'll call that RB. Then actually in what follows is the concentration of active ribosomes where there's no antibiotic bound. We call that RU for unbound. So there's a mistaken notation there. This should be RU. And then there's a binding rate. We've got K on. There should also be a K off binding rate. So this picture is a little bit inaccurate. And then also we've got an influx of antibiotic J and a growth rate lambda. So one thing here, we cheat, not quite cheat, but we avoid dealing with cell division explicitly. We just think of the cell growing, the concentrations diluting, and when there's an event where the cell divides, the concentrations are maintained. So we just follow the concentrations, and it's like following a single cell lineage rather than having to model a population of cells. Okay, so then we've got our three concentrations. We write down the simplest model, which is just differential equations for how these things evolve. So all the quantities here are simple. And I'll come back to the question marks in a moment. So lambda, remember, is the cell growth rate, and that dilutes all the concentrations, and then you can show you just get a simple negative term proportional to the growth rate. So just growing, you dilute concentrations, concentrations go down. F is the net unbinding, so that leads to an increase, because it's the unbinding of antibiotic, it will lead to an increase in the concentration of antibiotic and an increase in the concentration of unbound ribosomes, but a decrease, so a minus in the concentration of bound ribosomes. Then there's an external influx of antibiotic. So here we're going to think of fixing the external concentration. And finally, there's S, is the synthesis rate. And so what we need to think about, so that unbinding rate, you can, yep, okay. Unbinding rate is, okay, this is just mass action kinetics. We get a positive term from the unbinding of the concentration RB with rate K off, and a negative, okay, and a negative, oh, okay.
Just turn it off.
Right, have to get close. Yeah, so for the net unbinding, we've got a positive term from unbinding and a negative term from binding. The only thing to note here is that there's an offset arm in, and I'll come back to that later. And the idea is actually it's considered that the cell maintains a minimum concentration of unbound ribosomes. That's just, and we'll see that empirically. And then the influx is just in terms of permeabilities and concentrations. So this is just simple. The details actually don't matter. This is the important thing here. And so what's left is like the growth rate lambda and the synthesis rate. How do they depend on the parameters we've got and other parameters that we haven't included? Because these are more complicated processes, right, that are involving external stuff. And what we want to do is to use empirical data to model these, the growth rate and the synthesis rate. And to do that, we're going to go back to, these, what are called empirical growth laws. So the benchmark now is a 2010 science paper by Matt Scott in a group of Terry Roy. I mean, these measurements were first started, I believe, by Bremer and Dennis. And just to summarize these results, basically they're experiments, and as a physicist, I think of them as kind of fit. Is this working now? Okay. So the base growth rate, lambda naught, they measure that, and then they freeze the population and measure the concentration of ribosomes. So because there's no antibiotic here, that's unbound ribosomes in our language. And what they find is they get a linear fit with an offset, and that's the offset that I mentioned. So that you can think of that there's always some maintained a minimum population of unbound ribosomes. And the second measurement, which is different, is to fix the bacteria. So we're not measuring so we're not changing the base growth rate here. We're fixing that, but then varying the antibiotic. So then we're changing this growth inhibition, or this lambda as a function of A. And then they do the same measurement, but because it's antibiotic, there'll be both unbound and bound ribosomes that they measure, and measure that concentration. And again, they get a linear fit, but this time it's a negative coefficient for lambda of A. And these, if you think about it, these are not contradictory at all. They're two different measurements. Here you're varying the quantity lambda naught. Here, if you like, you're fixing lambda naught and varying lambda as a function of A. And so again, they get a nice linear fit, and this is some constant that, okay, I write it as a constant, it has some parameters there. So does that make sense? It means, yeah, okay, so if we add antibiotic, if you think about it, lambda should decrease, therefore because of the minus sign, the ribosome content increases. I'm not sure what I'm doing wrong here. Yeah, no, it's okay. Yeah. Yeah. It's okay. Okay, so what was I saying? Yeah. Sorry, back to this one. So, again, if we increase A, lambda decreases, therefore the ribosome content increases. And that makes sense because it's the thinking is that the bacterial cell rises to the challenge of the antibiotic and increases the production of ribosomes to counter the antibiotic action. Okay, so we're now going to use those two growth laws to close our system. So we're going to look at the steady state. So we put the time derivatives to 0. And what that means is it means that the concentrations are constant in time, but the population is actually growing in a stationary way. So if we put them all to 0, then we've got 3 equations. If we add 2 and 3, the F cancels, and we get S, okay, which makes sense. The synthesis rate should be the growth rate times the ribosome concentration. But then, we know from the empirical growth law that the total ribosome concentration is a linear function of lambda. So in the end, we get S as a quadratic in lambda. So that's the key thing. So just to show that it makes sense, so if we have S as a function of lambda, it would look something like that. So as lambda decreases a bit because of antibiotic, then the synthesis rate has to increase. The cell has to it gets upregulated to counter the antibiotic challenge. And then if we go down here, eventually S is 0 and lambda is 0. But then the interesting part is up here. Okay, and then, so that gives one more, okay, that gives S as a function of lambda. Then if we look at what we've got left, we've got lambda, A, Ru and Rb. So we've got 4 quantities, we need one more equation. And the second equation we can get from the first growth law. That says the unbound concentration is a linear function of lambda. So we're cheating slightly here, because remember this growth law was actually for lambda naught. We're going to assume it holds for lambda. Then we have 4 equations in 4 unknowns, and we can solve the equations. So if you do that, and this also, you can solve it, but it's a little bit simpler in the limit. K-on is very much less than this parameter kappa. Kappa is sort of the translational capacity. So anyway, okay, this is harmless. And then we have a fairly simple cubic equation. And the interesting thing, so this is just a solution to the system of 4 equations, and it's a solution for lambda in terms of lambda naught, the base growth rate.
Your kappa is dimensionless?
No. Kappa is it's translational capacity, so it's something density per unit time, I think. So it's got dimensions. It does, sorry, what's worrying you?
Well, I was just remembering back that you said the rate was lambda over kappa in the first law. So lambda had the same units as rate, but kappa would be dimensionless.
You are correct. Okay, so it's possible that kappa is not quite the same kappa. Okay, I need to think about that. It's a good point. I need to think about it. But for the moment, right, I have a cubic equation for lambda. So first, and the two parameters, we had quite a lot of parameters in the model, and they reduced to two parameters, lambda naught star and what we call the IC50 star, for reasons that will become apparent. So lambda naught star we call reversibility parameter, and basically because it increases with k off. Or if the ratio k off over k on is large, it's kind of reversible, so this is larger. And this IC50 is another parameter, and it involves those two offsets, R max and R min. So they're all wrapped up in one parameter, along with the inwards permeability. So great, we've got a cubic equation. If we're interested in the IC50, remember IC50 is the concentration at which lambda is reduced by a half. So we just set lambda over lambda naught to be a half, and then we solve for A, and that will give us the IC50. So we actually get a prediction here, and then you see that's why we call that parameter combination IC50 star, because it appears there. Okay. Yeah, sorry about this. And now the interesting thing here, as we'll see, is that this formula, the first part is an increasing function of the base growth rate, and the second is a decreasing function. So as we'll see, whether IC50 decreases or increases with lambda naught depends on which one of these dominates, and so it depends on the value of lambda naught star. So that's one of our predictions. I'll come back and we'll see that in more detail. Okay, so then it's a cubic equation, remember, for lambda over lambda naught. If we solve that numerically, okay, we've got 3 roots. So if lambda over lambda, what's the parameter here? If lambda naught star over lambda naught is low, we end up with 3 roots. And what happens is that we get a jump. Okay, so we increase A, we follow this route, and then we jump down to this one. So we get that discontinuous behavior. On the other hand, if lambda naught star over lambda naught is large, we just see one root. The cubic only has one real root, and we get a smooth curve. And that basically explains the two behaviors that we see experimentally. One can go further, and if we look at the limits of lambda naught star over lambda naught very small, if I go back, then in that case we would lose these terms here, and then this will actually become quadratic for lambda over lambda naught. And if we solve that quadratic, this is the relevant root. And what we see, so in this limit, what you see here is that you're coming down this curve here and then jumping to 0. So you get this discontinuous transition. On the other hand, if lambda naught star over lambda naught is large, then it's basically these terms, the quadratic terms, that will dominate. And we get a simple expression for lambda over lambda naught, which is similar to a monod law and has that smooth dependence. So these are two limits. And actually in these limits there's no parameters. So first of all is to fit the strep and can, remember that's the bactericidal data, we'll fit that to the first, actually there's no fit, that's the whole point. There's no, these formulas just involve the IC50, which is known experimentally, and lambda naught, which is known experimentally. So here there's actually no fitting parameters, we just try and collapse the data. So we see for the bactericidal, it's not bad, not perfect. As a physicist, I'm not so concerned because I know from experience with discontinuous transitions, you rarely see the jump cleanly. It tends to be smoothed out for various reasons. But these are actually pretty good, I think, for the bacteriostatic case. It's a pretty good fit. So then one can go further and go back to the full formula with the two parameters lambda naught star and IC50 star and try and fit all the data. So this is actually a challenge. Remember, you've got 6 data sets here, basically, and one is using just 2 parameters to fit, all the data sets. And again, the fit that you get is pretty good for the bacteriostatic. It's not bad for the bactericidal. But anyway, from this, you get 2 fitted parameters. So you might ask, well, we actually had formula for these parameters in terms of microscopic parameters. Why not just go and use the experimental values? And it's unfortunately out there, the experimental values are not precise enough. So we can't do that. Anyway, so these are the fits, and we fit these values of lambda naught star and IC50 star for each of the antibiotics and the media. So then the final thing is to collapse the data onto the universal curve. So again, for each antibiotic, we've got the 2, well, okay, so sorry, I should have said what I said wasn't quite true. For each antibiotic, we fit the lambda naught star and the IC50 star once for the glucose and once for the glycerol. We need to do an independent fit in each time, but for each one, there's 3 data sets that have to be fitted with the 2 parameters. Then if we collapse all the data, finally, because we've fixed our values of IC50 star and lambda naught star, then actually this is what we were most impressed with, that we get a pretty good collapse onto this universal curve. So as physicists, this is what we like, universality. We like to reduce lots of different things onto one curve. I have to say I have no problems with the referees for the paper, but I would say, they were very constructive, but clearly this was not the thing that interested them, and they were interested in other things. So it just shows there's a slightly different points of interest and sort of ethos between different disciplines. But this is the curve that we thought was the most significant, that the fact that you can collapse all the antibiotics onto one curve, and then where it is on this curve is essentially the value of the reversibility parameter. And so if you have a high or a low reversibility parameter, you get bactericidal behavior. If you have a high reversibility parameter, you get bacteriostatic. And then what you'll notice is that chloramphenicol can sort of creep across the 2 regimes. So that one's slightly ambiguous. Anyway, yeah. Actually, I've gone a bit quick. That's really what I wanted to say. So what the point was, the experiments showed a complex relation between the susceptibility to antibiotic, that's the growth curve, and the growth environment. And what we were impressed by that, we just wrote down, if you like, a very simple model just with 3 concentrations, and yet we did seem to get something that reproduced to some extent these growth curves. And there was actually a prediction that came out that the reversibility rate could provide a natural classification. And also if you could vary that, you could maybe change one antibiotic from being bactericidal to bacteriostatic. And it coincided with the normal classification of these 4 antibiotics into bacteriostatic and bactericidal. And what we wanted to do was to vary this translational capacity, kappa, by using mutants to try and vary that, and then through that vary lambda. This hasn't been so successful, I have to say. Otherwise, I think, yeah, that's all I wanted to say. Thank you.
I'll give it back.
Okay.
Yeah? Okay. Is that working? Okay.
Thank you for the talk. I was just missing, could you explain back again why you call it this lambda naught star, this reversibility rate? I missed, I guess, okay, the point of this name.
So, lambda naught star. So remember, we're solving the system of equations. The solution appears like this. So this is a parameter combination that appears from the equations. And what we say is that, okay, the factors involved here, there's a number of different factors. The rate of unbinding to binding is in there. So when that increases, you'd say it's more reversible, lambda naught increases. as P out increases, so this is the antibiotic going out, that also increases lambda naught. So again, you might think of that as reversibility characteristic. So all these factors feed into sort of one parameter, reversibility parameter.
Okay.
No, that was so maybe I didn't stress this enough. There's upwards of 6, 8 parameters. They all collapse onto these two, lambda naught star and the IC50 star, which again, as a physicist, that's what we like, to not have too many parameters.
Okay, a second question about this hysteresis effect.
Yeah.
Would there be any way to experimentally test the hysteresis effect? So you should have a different IC50 going the other way of...
Yes, that's true. Okay. So this is a good point. If we're going this way, the growth rate should, if we increase A from 0, the growth rate should go down smoothly. We get to there, it jumps, and then if we go the other way, it should... we've tried, okay, to be honest, we haven't been able to do that experimentally yet. We've studied it in the simulations. Because here I just showed you the stationary state, or the fixed points of the dynamics. One can look at the transients, and so we've looked at that, but we haven't looked experimentally. That would be a good thing to do.
Ça marche. I was wondering if the type of mutations that you observe when you treat bacteria with static or side drugs are the same, which or against asked differently, do they target the same? Do they affect the same mechanisms in the cell when you treat with bacteriostatic or bactericidal drugs? Or it's not clear?
No, okay, both of them here target the ribosomes or the translation process, but they're targeting different parts of the genome. So they do different things, but in the end they affect the protein synthesis.
Okay, but
that's
all different proteins.
Yes, yeah. So that's true. Okay, I can't give you the details. So we bundle them all up into just ribosome targeting. So our model is very coarse-grained, that we don't input exactly what they're targeting. We just say it affects the protein synthesis in some way, and we don't say what proteins it affects either. We don't get into that level of detail. That would be,
Yeah, because actually I was wondering about the way mutation will change the growth rate, the lambda, depending on the...
Yes. Well, our idea was to try and make mutations to change the translational rate of which the ribosome goes along. But that ended up being not very clean.
Yeah, thank you.
I have a question. Do you have the data for other kinds of antibiotics, for example, for the growth rate versus concentration of antibiotics? For example, for cell wall targeting antibiotics or...
No, not that I'm aware. Okay, I'm not the...
Because I'm just curious if you would see the same kind of
I understand your question. It's a logical question. We've done this for ribosome targeting. Does it also apply for other types of antibiotics? I actually don't know. I'd have to ask my colleague Matt.
So can you comment on the extensions to take into account phenotypic heterogeneity in growth rates? If I look at a population, it's often observed that not all the cells are growing at the same rate.
Yes.
And the ones that are growing slower are less susceptible. So could you extend this framework?
Good point. We haven't thought of that. That's an obvious thing to think about. Yeah. So phenotypic heterogeneity within the bacterial population. This is an idea that within a bacterial population, you have some guys who can survive the antibiotic attack, and so you have them as sort of in reserve, even though they're not so fit otherwise. Yeah. No, we haven't looked at that at all. Within this model, yeah. We've kept that to different studies completely. Yeah, that would be a good thing to do.
Yeah, just following up on what he said. So, like, also these are like mostly you're thinking of mean field. I mean, you are using a mean field approach, but just based on these ideas, I was having a conversation with Ariel Amir at Harvard. So he was kind of arguing that possibly the variability in growth rate is something that is tuned independent of the mean, and that is a possible approach for survival for bacterial colonies, for instance. I mean, it's more of a theoretical idea that he has. So I was wondering if you actually think of the distribution, I mean, next level, where you take into consideration the stochasticity and look into all these kind of, like, what kind of impacts you would have. Yeah.
Okay, as a physicist, this is the obvious extension. So you're absolutely right. The equations we write down, you can think of as mean field or else deterministic equations. They're simply ordinary differential equations which are deterministic. So if you make them stochastic, what happens? Philip Grulich, he tried this a bit. It became a big task. And we haven't pursued it fully. Again, sorry, it's not a very convincing answer. That's next on the agenda of things to do.
Bacterial persistence, come to my talk on Thursday.
Okay.
I'm going to talk exactly about that.
Great.
Thank you.
One question.
I think you need an assistant with the microphone. I have a question about this limit, lambda star over lambda small, which you write in the middle. No, well, you had it on the. I guess I can say it from the equation.
That one.
Yeah, it says limit, right? Wouldn't you still have three solutions still in that limit? That is, wouldn't lambda equals 0 be a solution, and wouldn't lambda equals lambda naught also be a, wouldn't there still be three branches, I guess what I'm saying.
Goes off to infinity. So we can look, I didn't. Okay, so if this, right, what I'm saying, yeah, okay, I see what you're saying.
You still have a cubic.
Yes, you're right.
But
okay, so
lambda equals 0 is a 1.
We can ignore this term.
Yeah.
So certainly we have a term solution lambda equals 0. Then you're right, there's a root of a quadratic. So one is the physical, yes, you're right, one is the physical and one is unphysical because it's dynamically unstable.
Uh-huh.
That's what we're saying.
I see. So you lambda equals 0, trivial, you'd say.
Yeah.
Then one's physical, and then one is non-physical. Well, it's unstable. Yeah. You wouldn't see it. Okay. All right.
Or I have to check that. Okay. Possibly it's negative even, so it's really unphysical. Okay, I have to think about it.
I would think it would just be the plus or minus radical.
Yeah. No, that's right. So
they both be possible.
Yeah.
Okay, so since Oana teased about a talk, I will also say that if you're interested in the mechanisms of antibiotic resistance, you should also listen to my talk tomorrow. So, okay, let's conclude this session, and thank Martin again. And
So before I start, I just want to make a comment. In many previous studies, for a smaller set of patterns, automated algorithms vastly outperform humans. So the difference here is it's a larger set of patterns, a larger range of numbers of proteins, but the previous 11 patterns suggest that there's hope there.
I think it'll improve a lot in the next year.
Yeah. So what I'm going to talk to you about, whoops. Okay, you broke it, Devin. switch around.
There was all that clicking I did.
There we go. Yep. All right. So we've heard of some fantastic work at the Human Protein Atlas, and we obviously are aware of the fact that fluorescence microscopy is great, especially for studying cell processes. But the problem is that we have 20,000 proteins at least, and we can't image in the same cell, especially in live cells, anywhere close to that number of proteins. And so we have to try to find a way to be able to take images of a smaller number of proteins at the same time in a live cell and learn something about all of the proteins by doing that many times. Right? So we need to try to find a way that we can use different images of different cells to build a coherent picture of what a whole cell is looking like and what it's doing. And so the idea that we've been pushing for a while now is that we can use generative models of the distribution of proteins as potentially this mechanism by which we can link information from different sets of images for different cells. And I'm going to talk to you about some variations on that theme, which is all about inferring what you can't directly measure, which is building off of the theme of estimating cell cycle from asynchronous cells that Devin talked about. And a particular context of this is that we would like to be able to build models that have many more proteins in them than we can directly measure in order to be able to do cell simulations with realistic geometries. So I'm going to start by telling a story about punctate organelles, which actually builds off of the images of the Human Protein Atlas. And the idea here, for this particular part, we're going to talk about comparing images of different proteins that have been stained in different cells, but try to find a way to relate them to each other through a common reference. And so what we're going to do is we're going to try to do that and also learn what organelles might another organelle depend on spatially. So we started out by taking images of 11 different proteins from the Human Protein Atlas. In this case, the human labelers are not able to distinguish the different classes of what they call vesicles, and so we asked whether we would be able to do that computationally. So these are examples of 11 different proteins that we know are different because of the protein that was labeled in these cells has been characterized elsewhere, both in vivo and in vitro, as being part of peroxisomes or being part of COP2 vesicles or whatever, right? So these are proteins that are, quote, known to be in different distinct structures, but visually they are all assigned to the same category. And so what we're going to do, very straightforward, is we're going to segment out the individual puncta. But notice all of these are small things that you could roughly think of as a puncta, that you don't have to really worry about so much their structure. So we're going to segment out the individual organelles, and we're going to segment out, it's hard to see in the red here, these are the microtubule stains that Devin showed. And we're going to have then an image of the puncta and an image of the microtubules, and we're going to use that to try to build a model of the positions of the puncta within the cell that is going to be dependent upon some other components of the cell. All right, and so that is illustrated here. So we're going to say this is a basic point process model. The idea is that the probability or the frequency of observing n objects, n of these puncta at specific positions x sub I, so that the simultaneously observing them at n distinct locations, is just the probability, the product of some function that is a function of the position of the puncta, right? Then do that over all n organelles and then normalize with a partition coefficient. All right, so this is very straightforward. The key is going to be, what's this all about? What's this function? All right, so what we're going to say is some aspects of the stele, we're going to try to find them that allow us to predict well the positions of the puncta that we observe. And yeah, sure.
X of I is a vector?
X is, it's the coordinates of three-dimensional coordinates or two-dimensional coordinates in this case of a puncta, one puncta. And so what we're going to do then is to say, let's explore things that we can measure from the images we have. And so the factors that we're going to consider are distance to the nuclear boundary, distance to the cell boundary, the density of microtubules at that point, the distance to the nearest microtubule, density of ER, and distance CR. I haven't shown the ER, but Devin already showed examples of that. Okay, so what we're going to ask then is which factors can we build the best model to predict the distribution, the spatial distribution of each of those 11 proteins. And we're going to do that by cross-validation. So what we're going to do is we're going to take half of the data, train a model, a point process model like I showed you, and then evaluate that point process model by calculating the predicted probability at the observed positions in a test image. And then that's the estimated likelihood of that test image having been generated by the model learned from the training images. So that's the estimated likelihood of that data given that model. Okay? And then we do that by cross-validation by changing around.
Can you pick these patterns because that's all that you had available in terms of information?
So these were all of the known, these factors. That's all we have available because the HPA images, they have a nuclear stain, a microtubule stain, an ER stain, and we can estimate the plasma membrane, by image processing. So, yeah. So, and then we're going to do that over multiple rounds of cross-validation and average. Okay? And so what we did then is do that. We did that first by building a model relative to the nuclear boundary, and then building a model with both the nuclear boundary and the cell boundary, and the likelihood went up from adding that second component. I'm summarizing the comparisons of all the different factors, but this is, and so the likelihood went up. What that says, it is a better model if you use both the nuclear boundary and the cell boundary to predict the positions of the puncta. If you add in the microtubule stain, the microtubule factors, the likelihood goes up again. So it says that's a better model also. But if you add in the ER marker, the likelihood actually goes down. That's telling you were overfitting. It was thinking that it was finding something, but it's not actually correct. And so the conclusion is that these are the 4 things you should use in order to try to build the best model you can for these images with what you have available. Now the question is, how good is that? And we can ask that as well by cross-validation. We can ask, can we use the model trained for the 11 proteins to apply to test proteins and ask whether the test protein is recognized as being from its correct model? Okay? Now, the problem here is this is, everybody, deep learning is the rage, right? This is the shallowest learning you can have because we have 2 images of every protein. So we use one of the images for training and we use one of them for testing, okay? And that's it. So, but then we get combinations. We can take, out of the 11 proteins, we can take different ones of each for each iteration in the cross-validation. Okay. Here are the results. The main point here is, here are the first time I'm telling you the different categories that we picked. Whoops. And you'll notice some of these things are not vesicles at all, which we sort of knew. They're things that, like RNP bodies, that aren't a vesicle at all, but they appear as if they're vesicles. The system can distinguish them well, right? So you see a lot of ones. The overall accuracies are here for 3 different cell lines, with 3 different training schemes, 3 different runs of the training algorithm. So I don't think there are any gamers out here that could tell the difference between those 11 proteins, but maybe next iteration Devin can check whether that's true. By the way, it's a generative model, and you can synthesize a hypothetical distribution of all 11 organelles in the same cell if you were so inclined. All right. Now, to build on that, what we're going to do is we're going to use the same point process model technology, but we're going to apply it to a different problem. And to try to illustrate the next step in this process, which is to learn how to put together something that is larger than you can measure at one time. All right? And so what we have here is a case where we're going to try to learn the spatial dependencies of components and then infer from the spatial dependencies what things are likely to be put together in what order in the cell. All right, and the specific case here is influenza virus assembly. Influenza has 8 ribonucleoprotein particles made in the nucleus, go out in the cytoplasm, and then form a virion where you need to have one of each of the 8 things. Okay? And the question is, what happens between here as they exit and here as you make a virion? What order, if any, do they get assembled in? And remember, you have to get one of each. So it's not like you can just sort of grab a bag of 8 of them. You won't end up with one of each that way. Right? So there has to be some mechanism by which this can happen. So what we're going to do is we're going to take 8, remember there's 8 of them, we're going to take images, not us, this is our collaborator, Simul Aquadalis did this, take images of 4 of the 8 at a time, and do various combinations of 4 of 8. Not all of them, that would be way too many, but all of the trimers and all of the dimers were done, right? In a set that included many of the quads, but not all of the quads, about a third of the quads. Okay. So we're going to build, as you can get, as I said, point process models. And what we're going to do is to learn those models to predict each one of the RNPs from as many of the other ones as we have it measured in combination with. So we're going to try to learn a model of it relative to each of the other 7 individually, or a model of it relative to pairs of the other 7, or those cases where there are triples, actually learn a model compared to that, and as well include the nuclear and membrane and plasma membrane, which we also have. Okay, and then for each RNP, we're going to pick the best model that we can train. So it might be that the best model depends on just one of the other RNPs, or it doesn't depend on the cell membrane, or it does. We're going to pick the best model that we can get that is predictive of the distribution of one of those RNPs. All right, so that's going to give us sort of 8 best models for each of the individual RNPs, and we also have models that tell us how to predict something based on 2 or 3 of the others. Okay. Now what we're going to do is we're going to use dynamic programming to answer the question, what sequence of steps would give us a full 8-mer in the most likely manner. So in other words, we put together these 3 and then we say, okay, with those 3 I can predict the distribution of this 4th one or this 5th one or for the 5th one, and I do that and I try all combinations of how to put that together, and I can form a tree that tells us what order I should put things together. Does that make sense to everyone? I know that's a little tricky, but I guess actually you guys are math people, so it's not tricky at all. Yes?
Your experiments are 1s, 2s, and 3s.
Up to 4.
Up to 4s. So you don't have to guess anything until you get to 5? I mean, not guess,
yeah. Well, some of the 4s are not present.
So you don't have all the 4s?
Don't have all the 4s.
Yeah.
But then you're absolutely right. Then you have to start guessing at 5s. Right.
Okay.
Yes. And so you're going to start inferring. But it might be that the highest likelihood 4 doesn't actually happen, because it might have been better to make a 2, and so on. Right.
Right.
So
but the likelihoods of the steps in the program are experimentally determined for the smaller bits, and then you start
and
to
then
use this
inferred for the higher ones, exactly.
Yeah. Okay.
Yes. Okay, so here, just to show you the differences, this is just the pairwise influences between the pairs, right? So if you have the skinny end is where it's coming from, so M predicts PB2 medium well, M predicts PB1 pretty well. Okay, that's just to see that there's variation among the predictions, which is, you know, you're going to need that in order to be able to do any inference.
So
Yeah.
the thickness is the relative likelihood.
The relative likelihood of predicting just from that one component. I can't really show you all the higher ones. You know, they get too hairy. Okay, and then just a sanity check, we can check whether for those quads that we've measured, how well the model predicts the quads with data that wasn't measured in the quad, right? And so it does a pretty good job for all of them. It turns out there is one outlier that is all the combination. It's PB2NA plus either PB1 and or NS. We don't know why that one's the outlier, but it's the rest of them pretty good. So still open question of why that's happening. We can also do the same thing with triplets predicting quads. Same thing, we get good predictions. Still that same outlier. All right, so we're going to now do the dynamic programming, and this is the tree that we get, which suggests that you put together these 4 proteins, then these 3 proteins, and then add in this one at the end. Now, we've got a lot of numerical calculation in here, so one of the first things to ask is how robust this is, how generalizable this is. So what we did is we added up to 25% noise to the likelihood of all of them, you know, randomly chosen with a standard deviation of 25%. And we got the same tree for all of those. If we want more than 25%, you started getting other trees. But that's a pretty hefty addition of noise, and you basically still get the same tree. The one other thing that we did in that respect is we removed those models that had directly measured quads, so that now everybody was on the same, because some of the quads were measured and some were not. And so now everybody's on the same playing field because we have all the triples, and we removed the quads. And the only thing that changes is that HA is moved from being added right away. This one says it's added right away. This one says it gets added a little bit later. All right, so it doesn't really fundamentally change anything in the.
How am I interpreting how am I interpreting the tree, right? So the bottom layer I get are the 8 things that we have.
Yep, and this says make this into a piece.
Into a piece of?
Into a complex.
Okay.
Make this into a complex. Well, let's go with that one. Make this into a complex. Yeah. Then join those 2 complexes and then add PA to the end.
Right.
And that's the most likely way to put this together. And that is reflected in the fact that these things can predict each other. So that means they have similar patterns. And these guys can predict each other. And then the ordering here depends on the higher order measurements. Okay? All right. And we actually went further. We removed the evidence, you know, the measurement of this triple, so that it was not allowed to be part of the inference, and it makes a very small change as well. So what the punchline from this part is we have a pretty consistent result with respect to the likely ordering of these influenza virus assembly, and a generalizable method for potentially inferring assembly of higher order complexes from lower order data where this type of paradigm applies. So I'm going to very quickly mention 2 more things. Well, this one quickly. And that is, again, with this theme of trying to infer what you can't measure. A common problem with fluorescence microscopy is the resolution. Even with super resolution microscopy, it's limited compared, for example, to electron microscopy. And so one technique that is used to date is to take images, so-called correlative light and electron microscopy, where you take the same sample, take a fluorescence image, and then take an EM image, fix it and take an EM image in order to try to learn a higher resolution structure for something that's labeled in fluorescence. Problem is that technique is very demanding and expensive, and so what we would be interested in is there a way that we can learn a model that will predict a higher resolution structure from a lower resolution structure or vice versa, okay? And so here's the approach that we took to this. The idea is that what we're gonna do is the starting data, this is the weird thing about generative models, right? You always start from what you don't know. But this is the starting data. It's EM images that we've segmented into two classes, of there's a nucleus and there's a membrane, okay? And then we learn a generative model that can start from random noise and generate a labeled image like that, or start from a labeled image like that and generate an image. Okay, was that clear? All right. So this, by the way, looks very much like it could have been a fluorescence microscope image. Right? So that's the reason we started with this. The idea was, could we potentially take a fluorescence microscope image and then apply this thing that we learned to predict what the EM structure might look like? Okay, this is showing the structure of the prediction. Where you're learning simultaneously to generate the labeled image and to generate the EM image, and of course the training works backwards. You're trying to produce images that are close to the real images. So here are some examples of X is the real data, and G of sub X of Y is the data predicted from the label image. And I think you can see that most of those are pretty good, at least to maybe a first order. Yes?
Difference between microscopical images and electron microscopy images, the fractional dye labeling that you usually have. So even if you go super resolution, you have these segments of non-emitting regions that you can't regenerate. You have to use some sort of a continuous function in order to predict the electron microscopy image from the segmented.
You're talking about tomography?
Yeah, for instance.
So these are 2D. These are all 2D right now.
Or using 2D with stochastical optical fluctuation imaging, for instance. Because you'll always have these segments of non-labeled regions in optical microscopy.
Sure. The issue here is not to get the actual resolution of an EM image, but to come as close as you can. Right, that is the goal here. This is just the higher resolution version of some of those, just to show you that they're pretty good at reconstructing the images. Yes?
Yeah, so here the X is used in training as well, or these are tests?
These are tests. These are test images.
So I'm trying to gather what, I thought there was a good example on the previous slide actually, because my initial thought is it's just learning a texture. But then if you look at the middle bottom one, this very large cell has sort of a complex splotch in it that's not maybe a simple texture, at least. Seems to be getting that too. So what's your explanation for exactly what's going on here?
The whole game here is, some kind of, one-to-one mapping and a potential many-to-one mapping, right? You're talking about significantly different spatial scales, but for this to work at all, there has to be something about something of a given shape or given relationship to other shapes that allows you to try to predict, right? And so, in other words, if everything that looked like that had totally different structure inside of it, it's hopeless.
Right. So it's learning that things that are green in the sort of input image are darker in the output image.
And learning substructure to them.
right?
Yeah, right. That's the part that I think is more interesting. But,
Absolutely.
yeah.
But it's not going to be perfect by any means.
So at some point, you're doing segmentation and classification.
Yes.
Right?
Yes.
And that's, you glossed over it, but that's very hard for these images. How do you validate that? Or, I mean, it seems hard to me.
Yeah. Kind of the point is that you don't have to, because the task is really to regenerate realistic ones like this. This is just how we're getting there, right? And so this could be gobbledygook, and if it helps you get to here, that's good, right? Now, it just happens that I'm saying that that looks sort of like a fluorescence microscope image, you know, but I'm not saying that that's a perfect segmentation. I'm just saying it's a representation that we can map to the real image, right? Or approximate the real image. Okay, so I think this is just a start. I mean, but I did think you might be interested in it because it's in the theme that we've been talking about of trying to infer something you can't measure. Last thing to touch on dynamics a little bit, and it's a follow on to what Devin was talking about, and that is to try to learn kinetics, in this case of cell organization in general, from a collection of static images that, in this case, the difference from what Devin was talking about is these are static images that were taken at different time points in a process. All right? And that process is going to be the differentiation of a cell type called PC12 cells that, in the presence of nerve growth factor, go from a roundish cell and differentiate into a neuronal-like cell in terms of their structure. And so we collected images of these cells, 3D images of them stained with a mitochondrial probe, and those were taken at different times after adding nerve growth factors to the cells. The problem here is these are very photosensitive cells. So you might want to say, oh, I'll just take a movie to understand what's happening during this process of differentiation with nerve growth factor, but you can't, because even taking one 3D image is enough, you know, in terms of taxing the photosensitivity of the cell. So the question is, if we can't do that, can we try to synthesize a movie of the whole differentiation process by taking images at different time points? Yes?
So is that true also for multiphoton imaging?
So so multiphoton imaging is mostly used when you have deep samples. There's really not any advantage here.
Just,
you know.
Working near IR, so you literally don't damage yourself.
Yeah, I mean.
You need to have a better accuracy of your XY layer.
Yeah. We didn't try taking multiphoton images. Yeah, that's the real answer. But I'm not aware of any experiments using multiphoton that have been successful for this type of problem. Okay, so what we're going to do now we're going to take a detour to talk about cell and nuclear shape modeling, which is going to be the main part we're going to focus on. And this is a reasonably well-studied field. A lot of people do measurements of cell and nuclear shape for assessing the effects of drugs or assessing the effects of siRNAs on cell morphology, cell processes. But most of those approaches are based on using discriminative models of cell and nuclear shape. Things like measuring the elepticity of a cell, or measuring, you know, the volume or area defect, or something that is purely descriptive or discriminative, and may work well to tell the difference between A and B, but that doesn't allow you to generate either an A or a B. And so we're going to, on the other hand, use a generative approach, as you've seen, as we've done throughout the talk. And we need to be able to do that with high accuracy. If we're going to try to build a generative model, we're going to now focus on the accuracy of the reconstruction as our primary concern. And so we're going to evaluate a bunch of methods. We're going to evaluate at least 3 methods here. The one we've used a lot before is diffeomorphic modeling. The idea is that you measure
you take each pair of shapes that you're interested in modeling, in this case 2 nuclei, and you measure how much work it takes you to morph one of them into the other, and that's a measure that you can then construct a distance matrix for all of the examples in your data set, and then you can find a low-dimensional embedding of that distance matrix as a representation of the cell shape space. So that's one method. Another is to deep learning methods, where you basically use something like an autoencoder, where you feed in images, train it in order to maximize the generation on the output side being the same as the input image. All right? So there's a lot of variations on those. And the last we're going to talk about is spherical harmonic models. The idea there is you have some 3D shape like this. You find a mapping of this to a sphere, to the surface of a sphere, to minimize or to maximize the agreement between the areas of the meshes in this thing and the spaces on the sphere. Okay, and I was not trained as a mathematician, so that's as far as I'm going to go as far as explaining that part. But it's, I mean, you can understand the basic principle there.
This is different than the Gauss map, is that right?
Yes.
Which
and
is the standard differential geometer tool, right?
Right. And so then once you have the spherical this spherical mapping, spherical parameterization, then you do spherical harmonic transform, and you have coefficients that allow you to do an inverse transform to get back to the sphere, and then from there back to the original. Okay, so I think I just said that. And you're going to use the transform for the representation. Now, there are a bunch of open source implementations of some version of this, and the major failure point for those is that they don't work well when the shape doesn't look like a sphere, which is kind of obvious. And so we tested various implementations, and here's an original image, and here are the attempted, spherical parameterizations of those cells, by other methods. Here is what we're calling robust SVARM, or SVARM RPDM, that is doing a very good job of doing the mapping. And as we'll see, it does a very good job even for fairly non-spherical shapes. Okay, so here's just the punchline in terms of the different methods. This is evaluated on a 3D HeLa data set that we made a couple of years ago. And what you can see is, if you compare for that data set, either for low numbers of latent dimensions, I left that out, right? Once you're projecting into the embedding, you have to choose how many dimensions the embedding is supposed to be. And this is sort of a low value, and this is a kind of a high value, but for all the methods, we're keeping that the same. So they all either get 7 or 100, so that we're comparing apples to apples. And what you can see is that the robust spherical harmonic approach has the lowest reconstruction error for all of these different data sets, regardless of the number. And actually, the diffeomorphic methods sort of give up at higher dimensional mappings, and so we didn't even test them. So these just show you now the resulting two-dimensional projection of the embedding for cells that were exposed to NGF for different amounts of time. Okay, so blue means 0. The warmer the color, the later it is in the time course. All right, so these are the first 2 dimensions of shape. We've taken out orientation and size, so that's just shape, true shape for those who work in this space. And what you can see here is that this is just showing the trend line of going from blue to red over the time course of the NGF treatment. All right, so that is largely just change in this first principal component. Of course, there may be things happening in other dimensions we're not seeing, but but this is just kind of a simplification. However, the other thing you can see is, look how variable the blue ones are, and in fact all of them are. Right? So there is a lot of variation in the populations that even before you add the NGF. This is if we display just the first principal component versus size, and what you can see is that they're actually changing size as well as undergoing some undefined shape transformation, which seems to be sort of making these, spiky things. Okay, so that is the starting point now, and what we want to do is we want to try to imagine what's hypothetical trajectories in that shape space might look like. We have 100 cells for each of the different time points. So what we're going to do is we're going to make a single shape space like I showed you, and then we're going to assume that for each cell at the initial time point, there is a cell that could have been that same cell at the later time point. All right? It's actually a different cell, but it could look like what this cell would have looked like at the later time. So we're just going to match all of the cells up so that each cell gets matched to the cell that it would best match to in the next time frame, and the next, and the next, and the next, with an overall optimization, right? So that it's not greedy. What that means is that we essentially have 100 pseudo trajectories, right? 100, sets of images where we could imagine that we've imaged the same cell at all of the time points. Okay? So now we can interpolate in that with the generative model through that space in order to generate hypothetical transitions. So here are just some of those linked trajectories. This is the time with treatment of NGF, and these are deciles of similarity among the trajectories. Okay, so in here it's kind of the average trajectory. Here's one extreme, here's another extreme. All right, and it reflects the fact that there was heterogeneity in the initial time, and as we intended, the ones that are most dissimilar here from the average also end up being dissimilar here. Right, we're sort of following the bottom and the top of the transition, if you will. Does that concept make sense? Yeah?
Yeah, makes sense to me, but my question is, how do you estimate the sampling that you need in order to build an accurate trajectory, right? Because if you just had start and end point, you'd be totally guessing. And at some point, you don't really need more sampling, so there's a trade-off.
I wish I had an answer for you. I don't. I mean, we had one data set.
But you could drop out.
Oh, you mean drop it? Yes,
some
we could,
points and then see how close you get to it.
Yes, you're absolutely right. Yes. From that way, we can estimate the intermediate.
It doesn't tell you, because you don't have any time series on this, right?
There are no time series.
So it doesn't tell you whether you are accurately going through a trajectory, but at least tells you whether you're staying within the space of observable.
So we have done this, which is to measure how similar this guy is to the morphing of that into that.
Right.
Okay.
Oh, that's true too. Yeah, that's a good idea.
And that actually, they get quite close. Right? So in other words, as you're going through the shape space from one to the other, do you pass the one you skipped? And the answer is yes. Right. But I thought you were asking the question, how do I know how many cells I need in order to be able to reconstruct that? And I can only do down, and we haven't done that.
Yeah. Okay.
So just to finish up, here are movies, synthetic movies of the process starting from the beginning and differentiating to the end. Color now just is the major axis of the cell. It's not anything else. It's not depth or anything. But those, to me at least, look like reasonable interpolations of what the change in shape might be for these cells as they divide. Okay, so, and it gives us this hypothesis, which is sort of circular, but that the heterogeneity of the outcomes, which is quite significant, probably is coming from the heterogeneity of the input. But we can certainly try to test that at some point. All right, so overall, the bottom line is that these have been all examples of trying to do more than just analyze or compare images, but rather to take images and build models from them, that we can do some kind of inference to find information that we didn't have directly in what we started from. And I will stop there, and these are all the people that did the work. And Devin's on there somewhere. And thank you very much for your attention.
That was interesting and really nice. So I got a little lost at the end, but maybe I can ask a question for clarification. The matrix you had of images at the end there showed kind of intuitively size and extension or something like that. Does that explain the effectively 2 dimensions that were coming out of the PCA analysis? Or no?
I only showed 2 dimensions.
Mm-hmm.
The model was actually much higher. It was a 100-dimensional model.
Yep.
That the inference is done in that space, but I'm only showing you 2 dimensions because I didn't know how else to do it.
Sure. I see. So you kind of forced it into 2 dimensions, you'd say. Yes. But you did have a plot showing, I don't know what it was, cell size versus 1. And that showed, in fact, that you're missing another variable, right? Is that just because it was from those 2 dimensions down to 1 and that you're just picking up the other one? Is that okay?
Exactly.
I see.
Yeah,
okay.
Yeah. I mean, the movies really are the main way that, because they are in the high dimensional space. So those, are a more accurate reflection of what's going on.
So, just a question of curiosity. In the first part of the talk, you presented this formation of the influenza capsid by showing the sequence. And I was wondering what is known about the biophysics, how the selection occurs, and also how can such a sequence of events occur one after the other? Is there any documentation in the literature after what you have shown,
So,
what you have presented?
Yeah, there has been some work on this problem, and what has been ruled out is some high affinity process where they get shipped out of the nucleus and immediately, form a trimer or something, and then the other ones form a tetramer, and then those 2 things, So there are not cleanly observable states like that, which suggests, and experiments also indicate, that the affinities between the individual particles are not that high. And so it is a probabilistic game that they are playing in, bumping into each other and sticking long enough that the next guy can come along, and then you get something that has a chance of bumping into, the thing that and that's the process. Something along those lines is the process, rather than a very high affinity, binding that would really drive it into distinct intermediates that you would be able to observe.
I'm curious about one point, and that's could you build a discriminative model that could discriminate between a cell and its mirror image? Because there are certain cells that a human observer could easily say that's the real cell, and the mirror image is the fake cell, and but those are very specialized cells that have a gross anatomical chirality.
Yeah.
But you don't often see this concept in, let's say, mammalian or human cells. But I wondered if, in fact, your methods could reveal that there's a hidden chirality in let's say, the mesoscopic arrangement of organelles that in fact has gone unnoticed by human observers.
So that's just a standard shape space question, right? I mean, in other words,
Yeah.
when, if you're doing alignment of the shapes, then chiral shapes face different directions after you've done the alignment, and so you would find two different populations. We don't see any evidence of that, any of these, but I didn't really expect it. But there may be some specialized cell type for which that would be the case. Given that these are, images on a flat surface, I think it's highly unlikely that that would happen, that you would actually have handedness in such a space. But who knows, All right.
Okay, let's thank the speaker again. And yeah, you're on your own.